Hi, my name is Becky Wells. I'm the co-director of CEMB, and formerly I was the director of education. Now as co-director, I also oversee the education programs, although honestly with Annie, there's really not very much to do. But so I want to welcome you all to what I realize is the 5th CEMB undergraduate symposium, which is pretty amazing to think about. And I want to thank you all for your hard work and enthusiasm over the summer. So I thought that a good way to end your 10-week experience would be to tell you about something we probably should have told you about at the very beginning, and that's the name of this program, because this has to do with our basic philosophy, and I'm hoping that you've all come to appreciate it. So when CEMB began, we referred to this as the undergraduate program, or using the National Science Foundation shorthand, the REU program, referring to the formal research experiences for undergraduates programs of the NSF. And the problem became that CEMB has yearly site visit evaluations that are led by a panel of experts convened by the NSF, outside experts. And one year they actually gave us quite a bit of grief for not running our program the way they wanted REUs to be run. And specifically, they didn't like it that we limited the program to rising juniors and seniors for the most part, and to those who already had basic research experience. They wanted us to include younger students, such as rising sophomores, and potentially even incoming freshmen, and to focus more on teaching the basics to students who had no research background at all. And the thing is that we actually really liked the way we ran our program, and we felt it served a very specific and important purpose. And that purpose was to push students to carry out integrative science. So CEMB as a center draws faculty and trainees from both the physical and the biological sciences. And one of the major goals of the center as a whole, as well as the undergrad program, is to develop scientists who are comfortable working and collaborating at that interface. And so we came up with the name very intentionally of UEXB, or Undergraduates Expanding Boundaries, to capture the concept that we take the best students, the very best students who've already been exposed to research in one area, and then we push them out of their comfort zones into more integrative science. It won't have worked for every single one of you, but I hope that many, if not most of you, got here and had an uncomfortable first couple of weeks in the lab, that you were being forced to do something that was out of your comfort zone, out of your box, not what you expected and not what you were used to, but that after the first couple of weeks, something clicked, and you realized that you could actually do it, that you were good at it, and that you produced great science in a way you might not have thought yourself capable of. So I want you to leave this program thinking about how we might have pushed you to think and work outside of your own box, and to think about the benefits you might have gotten from that approach, and to think about how that might apply to the rest of your career. Because I think that all of us in CEMB at all levels are sort of committed to this concept of trying to push ourselves beyond our own boundaries and outside of our boxes. And we feel that that's had great benefit to all of us individually and as a center as far as the science. So I'll leave it at that, but I hope you think about it. So thanks, Annie.
Great, thank you, Becky. And I suppose I should introduce myself for those who don't know. My name is Annie Jeong. I've I guess been like the director of the UPenn site for this program. And so today I'll serve as the moderator for the talks. And so to jump right in, we'll start with our first talk by Mística Lozano Perez. She is an undergraduate at the University of Texas at San Antonio, and this summer she worked in the Boerckel lab.
Hello, everybody. Can everyone see my screen?
Yes.
Okay, so let's jump into it. Good afternoon, everybody. My name is Mística, and over summer, I had the privilege to work alongside Dr. Anna Marie Lang in Dr. Joelle Boerckel's lab, where we conducted research related to bone regeneration. Now, everyone present today, hold on, my slides aren't moving forward. Okay, there you go. Now, everyone present today has either broken a bone themselves or knows somebody who has broken a bone. To understand the research I was a part of over summer, we must first understand how a bone heals after fracturing. When a bone undergoes a fracture, it will have to go through different stages to fully heal. It starts with an anabolic stage, which is characterized by low oxygen levels. And then when the oxygen levels begin to rise in the fracture area, the regeneration stage shifts to a more catabolic environment, meaning a more oxygen-rich environment. For the sake of today's talk, we will focus on the anabolic process of this regeneration process. The reason there are low oxygen levels in this phase is because the fracture causes a rupture in the blood vessels in the area, and thus removes the way oxygen can actually reach the affected area. In order to adapt to the low oxygen levels, cells go through a process known as the HIF pathway. The purpose of this pathway is to help stabilize the low oxygen levels by aiding the process of blood vessel regrowth. Under normal oxygen levels, also known as normoxia, which is a mix between normal and oxygen, enzymes known as PhDs attach an oxygen group to the HIF alpha protein that then allows the BHL protein to attach to HIF and ultimately degrade it. Go forward. What? Sorry, this is jumping for some reason. Yes. However, under low oxygen conditions known as hypoxia, the PhD enzymes are inactive and do not oxygenize the HIF protein, thus allowing the HIF protein to move into the nucleus and start the transcription of its target genes. As the HIF pathway responds to low oxygen levels, there is another pathway that activates in response to mechanical cues that act on the fracture site as it goes through the process of regeneration. This pathway is known as the HIPPO pathway. In this pathway, there are two downstream regulators known as YAP and TAZ that regulate cell proliferation and differentiation. The pathway works opposite to mechanical stimuli. So when there is little mechanical stress, such as tension in the cell, the HIPPO pathway will activate and prevent YAP and TAZ from entering the nucleus, thus preventing the transcription of cell proliferation and differentiation genes. However, once tension is actually added to the cells, the opposite will occur. So now the cells can actually sense the mechanical stimuli and react by turning off the HIPPO pathway, thus allowing YAP and TAZ to enter the nucleus and start transcription of cell differentiation and proliferation genes. Now, both of these pathways are very important because as the HIF pathway works to rebuild blood vessels to provide oxygen to the fracture, the YAP-TAZ regulators of the HIPPO pathway work on differentiating and proliferating cells to rebuild the bone and tissue at the fracture site. Both of these pathways start independently of each other. However, since they work parallel to one another, it led us to question whether they actually work independently or if they interact with each other while going through the regeneration process. In order to answer this question, we first had to set up and test some preliminary experiments with periosteal cells. These cells were harvested from mice periosteum, meaning the outer layer of bone and their leg bones. The first of these experiments is an osteogenic differentiation experiment that tests different treatment groups over the course of 2 weeks in both oxygen-rich environments, normoxia, and oxygen-starved environments, also known as hypoxia. This first graph shows all of the different testing groups along the x-axis, with the legend to the right that actually explained what each group was either inhibiting or stabilizing. These samples were kept in an oxygen-rich environment, and the samples for each time points were taken at either 7 days, which is shown in gray, or at 14 days, which is shown in pink, via an Almer Blue assay. The purpose of the Almer Blue assay is to quantify the cell metabolic activity, which could give us a relative amount of healthy cells in a single group. The Almer Blue assay does not give us a cell count itself. It kind of just tells us the cell metabolic activity. So while some cells will have a high metabolic activity, that does not necessarily mean that there's a lot of cells. So over the 2-week experiments, the group that showed the most drastic change in metabolic activity was the group treated with DFO, which is a chemical meant to stabilize HIF. It works to stabilize HIF by binding to the PhD enzymes from earlier and allow HIF to move into the nucleus to mimic the reaction of a hypoxia or low oxygen environment. Although the other treatment groups are important to the experiment, for the continuation of this talk, we will only be looking at the DFO compound. The previous graph showed the Almer Blue readings in a normoxic environment, but we also performed the exact same experiments under true hypoxic conditions to make sure that the cells did not get oversaturated with HIF. As seen in the graph, we found that HIF affects cell metabolic activity similarly to how it affected the cells in the normoxic conditions. In summary of experiment 1, we found that out of all of the treatment groups, DFO causes the most metabolic activity changes. As you can see in the pictures, the end goal of using DFO is to mimic hypoxic conditions through a chemical rather than an environment. Since stabilizing HIF with DFO showed the highest fold change in metabolic activity, we decided to design the next experiment to test the metabolic activity of cells exposed to gradually increasing concentrations of DFO. Because since we knew DFO affected it, we kind of wanted to see at what point it starts to actually harm them, or at what point we can use the least amount of DFO and still get the same results. By setting up a plate as shown in the picture, we were able to perform an almond blue assay after 3 and 7 days to measure any metabolic differences seen at a shorter time frame instead of 7 and 14 days. Each well was seeded with the same number of periosteo cells. The first well acted as the control with only osteogenic media and no DFO. Every blue dot is a nucleus stained with DAPI, and the yellow is an actin staining that stains the cytoskeleton. The second well has the least amount of DFO, and as you can see, there are some visible changes in the actin staining. As the DFO concentration increases, the cytoskeleton continues to show changes, until finally, at the last 2 wells, the differences do not appear as obvious. From these pictures, we can see that the cell cytoskeleton is somehow affected by DFO, but we don't really know exactly how, but we do see a difference, so it is a good starting point to move forward. In addition to testing different DFO concentrations, we also had to test if the seeding density of cells could affect the cell metabolic activity, similarly to the changes we saw in the very first experiments. This was important to test because we know that YAPTAZ is affected by cell tension and mechanical cues, but we also wanted to see if the readings we obtained in the previous experiments could be due to cell death instead of overproliferation. To test this, we seeded the cells at gradually decreasing concentrations in the 24 well plates, either in osteogenic medium or expansion medium. Osteogenic medium is designed to cause the cells to differentiate, while the expansion medium is just to let them actually grow in number and not differentiate. After 7 days, the sample was tested with an Elmer blue assay and then stained with DAPI and actin. By using the DAPI stain, we were actually able to get a relative cell counts for the amount of cells in each well. The graph you see has the different seeding dilutions along the x-axis, with 20,000 cells per centimeter squared acting as the normal seeding density and decreasing by a factor of 2 for the next 2 seeding densities. From the graph, it appears that a seeding density of about 10,000 cells per centimeter squared caused the most cell proliferation after the 7 days. This could be true, but we also have to remember that the normal seeding of 20,000 cells per centimeter squared could have gotten too full and just caused the cells to lift off of the plates. So even though the results do show one conclusion, there could be multiple reasons to this happening. All of the results we got from these experiments are good starting points for future experiments. For example, we can start to test further with other treatment groups seen in the very beginning, or we could even try to test the mix of DFO concentrations with different seeding densities. At this point, the possibilities are just about endless. Before I officially end this presentation, I would just like to touch on a very important topic that I learned about over summer, and it's about mice handling. Due to them giving their life for research, I learned that there are ways to treat them in a more humane manner than just tossing them in the cage. Through acts of tunnel handling, cage enrichment activities, and honestly just monitoring their overall body language for signs of pain or discomfort, we can show the mice that we appreciate their sacrifice and that they deserve to be treated with care and in a humane way. Thank you for listening, and thank you for an amazing summer.
Great, thank you. And if anybody has any questions, please feel free to unmute yourself or write your question in the chat and I can repeat it for you. All right. Well, if there are no more aren't any questions, thank you so much, Mystica. That was a great job. Next up, we have Deanna Cruz Garcia from UC Merced, and she spent her summer working in the Mauck lab.
Okay, hi. Can you guys see my presentation? Okay. Okay. So, hi everyone, my name is Diana, and I'll be presenting my summer research project titled Role of Actin Stress Fibers and Rho Signaling on Chondrogenic Gene Expression in ATDC5 cells as a function of substrate stiffness. And I worked on this in Dr. Robert Mauck's lab with my graduate student mentor, Ryan. So to start off, articular cartilage is what provides the smooth surface for compressive resistance and near frictionless movement. Articular cartilage is found at the end of the bone and connects to form joints. Healthy cartilage is what allows us to move easily, and this is composed of cells called articular chondrocytes. In healthy articular chondrocytes, there's physiologic loading, so there's an increase in extracellular matrix anabolism and a decrease in inflammation. A healthy chondrocyte would take this sort of morphology, a very round and small cell with actin labeled in red in a cortical-like organization, and the microtubules shown in green will form this mesh-like cage around the nucleus. It is important to note that the actin does not come into contact with the nucleus. Additionally, there is expression of chondrogenic markers such as SOX9, collagen 2, and aggrecan, versus in a injured chondrocyte, there is an increase in extracellular matrix catabolism and an increase in inflammation as well. And an injured chondrocyte will take up this sort of morphology, so it's very spindle enlarged type, and the actin is now actin stress fibers, which do come into contact with the nucleus, and the microtubules do not form this mesh-like cage around the nucleus anymore. And there's also a decrease in the chondrogenic markers of SOX9, collagen 2, and aggrecan. And this comes into importance due to mechanotransduction, which is the process by which cells convert biophysical stimuli into a biochemical response. And this happens when there's a tug or a force on the extracellular matrix, and this causes for actin to form, and this causes the nucleus to stretch and the nucleus pore to widen, and the transcription factors become more permeable to go inside the nucleus and alter gene expression. This cytoskeletal nuclear mechanotransduction is largely mediated by the link complex. And due to the scope of this summer project, I will be focusing on actin and not so much on the microtubules or the link complex today. And another important regulator of chondrogenesis is the Rho pathway. This pathway is essentially responsible for the production of actin. So what happens is when Rho activates ROCK, it activates coughlin, which stabilizes actin. And not only does activation of Rho and ROCK regulate actin stress fibers, but it also regulates contractility. Increased Rho signaling decreases chondrogenic gene expression, and this is thought to be due to increased actin stress fiber formation. Given that, I hypothesize that increased Rho signaling decreases chondrogenic gene expression in an actin-dependent manner. So I think if we are able to add actin stress fibers in a healthy chondrocyte, we are able to mimic a disease-like cell, and vice versa. If we have a cortical actin in a disease cell, we could mimic it to go back to a healthy state cell. To do so, we used ATDC5 cells, which are a chondroprogenitor cell line, and we seeded them on polyacrylamide gels on different substrate stiffnesses, such as 5 kilopascals, 55 kilopascals, and glass. We then cultured these cells using CM+ serum-free chemically defined media with TGF-beta3 to max out chondrogenic differentiation. We also conducted qPCR. We did this by isolating RNA samples from these cells on the PA gels, and we quantified gene expression of SOX9, collagen 2, and aggrecan. So one way to view the actin on different substrate stiffness is by using 2D hydrogel cultures, such as this study. They seeded cells on different substrate stiffnesses, varying from 1 kilopascal gels, which are very soft, versus very stiff substrates such as glass. And what they were able to see here, given the images, is as you increase substrate stiffness, there's an increase in cell spread area, an increase in contractility, and an increase in actin stress fiber formation. We wanted to try to do this ourselves, and what we were able to see was as we decrease substrate stiffness, there's a decrease in cell size and stress fibers, and thus the cells become more small and round. So in these images, F-actin is shown in red and DAPI in blue. And so on very stiff substrates, again, the cells are more elongated with a lot of actin stress fibers versus on very small PA gels, such as the 5 kilopascals, the cells are smaller, rounder, and the actin is more cortical-like. And this 55 plays as our intermediate between the two. So not only did we want to see that they exhibit different morphological differences based on substrate stiffness, but we also wanted to see if they were expressing the same chondrogenic markers. And what we saw was that there's actually an increase of chondrogenic gene expression on softer substrates compared to cells on stiff substrates. So on these graphs, there is a significant increase in chondrogenic gene expression on cells on soft gels versus cells on very stiff gels, and this is true for all 3 markers. So looking at using our hypothetical model, we were able to see that as you increase substrate stiffness, a healthy state cell can mimic more of a disease-like cell. So there seems to be this inverse relationship between substrate stiffness and chondrogenic gene expression. And so we wanted to see if we could use Rho and actin stress fibers on a healthy state cell to get it to mimic a disease-like phenotype and gene expression. And we tested this by using LPA, which is a Rho agonist, and jasblinkenolide, which is an actin stabilizer. And we actually were able to see this because when we increase activation of Rho signaling using LPA and increase actin stress fiber formation using the JSP, we saw that there is a decrease in chondrogenic gene expression. And in these graphs, you can see that there is a decrease in the cells on the gels, with LPA and JSP compared to the ones on 5 kilopascals alone. And this was true for all 3 markers. Here, we just wanted to see the qualitatively, like how the cells looked. So we saw that as we increase actin stress fibers, it seemed to also increase focal adhesions and contractile and cell spread area. So on this glass, on the cells on the glass, there seemed very elongated, large focal adhesions and a lot of actin stress fibers versus on this soft gel, the cells are smaller, rounder. Also on the 5 kilopascals with LPA, there seems to be a split between cell types. There's small ones and there's also elongated ones. And on the gels with JSP, there's larger focal adhesions. There seems to be less actin stress fiber formation. So circling back on this hypothetical model, since we were able to get a healthy state cell to sort of mimic this disease-like cell, then we wanted to see if we can get our intermediate gel, so plate cells on an intermediate gel and try to reverse it and get it to a more healthy state cell. And in order to test this, we used CTO4, which is a Rho inhibitor. And this is exactly what we saw. So decreasing Rho signaling using CTO4 led to an increase in chondrogenic gene expression. So on the gels with CTO4, there is a significant increase in gene expression compared to those on the intermediate gel without the CTO4. And we were able to see this with SOX9, collagen 2, and aggrecan. Similarly here, we wanted to qualitatively see the difference in morphology. So as we decrease Rho signaling and decrease cell size, actin stress fibers, and focal adhesions. So compared to the glass, the 55 kilopascal seems to take up the same morphology and has smaller focal adhesions. And the 55 kilopascal gels with CTO4, there seemed to be a bit more spindle-like, spider-like morphology with less actin stress fibers and smaller focal adhesions. And these are just our preliminary epifluorescent pictures. So in conclusion, our results suggest that increased Rho signaling potentially through actin stress fiber formation decreases chondrogenic gene expression. Also, increased Rho signaling could be a potential initiating mechanism in chondrocyte dedifferentiation in disease. As for future directions, we're interested in IL-1 beta, which is a major osteoarthritis regulator. And IL-1 beta increases actin stress fibers, cell stiffness, and it decreases chondrogenic gene expression. So it would be interesting to see if we could negate the effects of IL-1 beta by possibly co-treating it with CTO4. However, since CTO4 is not FDA approved, another possibility is to co-treat it with Fosdil, which is a ROCK inhibitor that is FDA approved for the treatment of ALS. So this could be a possible clinical study that could be done in lab. And lastly, I just want to acknowledge Dr. Mauck's lab, everyone in the lab for being so welcoming, and my mentor Ryan, which has been the best mentor, has taught me so much, and CEMB for giving me the opportunity to conduct research for the summer. Yeah, thank you.
Great, thank you, Dee. If anybody has questions for Dee, feel free to unmute yourself or put it in the chat.
Yeah, can I? Yeah, so Dee, that's great. Really excellent work and presentation. So I have a question I mean, you looked at one gene, right? So my question is, you know, we have a couple of preprints on the archive with Mauck Lab where they have looked at net amount of heterochromatin versus euchromatin, meaning overall gene expression as a function of stiffness. Did you or others in the lab look at overall gene expression as opposed to expression of one particular gene as a function of substrate stiffness?
No, I didn't, but I know my graduate student mentor is looking into that. I think just for the summer, for the scope of my project, I just focused on these 3 genes.
Okay, all right, thanks.
Yeah.
Great, thank you so much. So our next speaker is Noah Kim. He's a student at Auburn University, and he worked in the Shenoy Lab.
Hello, can everyone see my presentation? I'm not sure if I'm sharing the right screen.
Yes.
Okay, great. Yeah, so my name is Noah Kim, and I am from Auburn University, as Annie said, and I worked under Dr. Shenoy's lab at the University of Pennsylvania this summer. And I'll be presenting on a computational analysis of inertial loading on the brain. Okay, so my outline will be pretty simple, just the background of my work and kind of the theoretical details of that. And then I'll be going over my methods and results that I went through over the summer, and I'll take questions at the end. So the brain gets injured in various ways, and the type of injury that I'm studying this summer is, that I did study this summer, is diffuse axonal injury, which is related to damage in the neurons. And so a common way that neurons can get damaged is when a force is distributed along the axon, causing a shear deformation in the axon body. As you can see from the diagram, the axon's being like twisted, and that's causing it to break apart. And so this deformation causes cell degeneration or even death, and that can lead to a variety of neurodegenerative diseases that we do not want. And so because this type of damage is on a cellular level, you're looking at the neurons of the brain. Conventional ways to study this in lab is kind of difficult. And so that's where a computational simulation of brain movement can help. And so that is kind of why I'm doing my research. But in order to get correct results with the simulation, the brain model that you use needs to include key features that affect how forces distribute throughout the brain. And so one such feature would be the white matter in the brain. And so, as most of you probably know, the brain is composed of gray matter, which is made up of the cell bodies of neurons, and that is primarily homogeneous in material. So when you apply a force to the gray matter, it'll distribute pretty evenly, and so there's not much issue there. But the white matter in the brain, on the other hand, is made up of these elongated axons of neurons that stem out from the neuron bodies. And so these axons will arrange to form bundles that point in a certain direction, and that introduces anisotropy in the white matter, which is basically just a characteristic where properties of the matter will differ based off which direction you're looking at. And so a force distributed along the axon will travel differently than a force distributed parallel to these axons. And so just to kind of show you this, if you zoom into the white matter, there'll be different tissue fibers. And if you zoom into those fibers, you'll see these bundles that are made up of individual axons all facing in the same direction. And so the behavior where axons deform and shear is common in white matter due to this, these bundles and this anisotropy. When forces distribute throughout the white matter, adjacent bundles of these axons will rub together and cause a shear strain commonly seen in diffuse axonal injury. And so it's really important to include these axon traps in your brain model. The reason I'm emphasizing shear strain so much is that it has a much larger impact on diffuse axonal injury. And previous studies have actually observed this. So there are various criteria that you can use to judge white matter brain damage, but shear strain has the most effect. So you see more red on the left picture than on the right. And that's because they're looking at the shear strain in that diagram. And then the other key feature I was looking at over the summer was the ventricular region. And so the ventricles are just cavities in the brain filled with cerebrospinal fluid. And because of this difference between the fluid and the white matter surrounding the ventricles, there will be more force distributed around the ventricular region, especially at the corners. And so you can see in this ventricle model, the middle of it has a really big red spot right at the corners of all those ventricles in the middle of the brain. And so we also need to model this to get a correct brain model. And so moving on to the methods of my research, I got all of my brain data this summer from diffusion tensor imaging, which is a technique that uses magnetic resonance imaging to track diffusion of water molecules in white matter. And so what happens in diffusion tensor imaging is that water molecules will diffuse faster along axon tracks rather than against them. And this difference can be detected by applying magnetic fields in various directions. And once all these different directional magnetic fields are overlaid on top of each other and color-coded, you can get the color map that you can see on the left of the screen. And these different colors just represent different directions of the the water diffusion, which represents the axon tracks. If you zoom into a point on this map, you can see that the image is resolved into 3 units of space. And so in the middle figure here, 1 unit of space would be a 1 millimeter cube, basically. And inside of each cube are actually all the individual fibers. And since they're all basically pointing in the same direction, you can see they're all very parallel to each other. They get averaged and represented by 1 vector in each unit of space. And so the first half of my summer was spent using MATLAB to analyze these vectors from a diffusion tensor imaging experiment done at Stanford University. And so as you can see from this zoom up, there were areas with multiple vectors that needed to be resolved because sometimes the diffusion tensor imaging experiment will collect some faulty data or gather more directions than are actually there. And so to clean up this data, I created a MATLAB algorithm to just get rid of or reorganize some of the extraneous vectors. And so this looks a lot cleaner. And I was also able to use the data to get a brain outline for the simulation. And so once I had the brain model, I was able to move on to setting up the simulation. The first thing to do was to set up the type of movement we would simulate. And what we chose to simulate was rapid rotational acceleration and deceleration of the brain because it has a much more pronounced effect on axonal damage in white matter. These images from a previous study compare translational to rotational movement when observing white matter, and translational acceleration had essentially no effect. So modeling rotational acceleration was what we did this summer. And so to do this, I used COMSOL Multiphysics, which is a physics simulation software to model this rotational impulse. And so this graph at the top shows an interpolated curve from an experiment done on a swine brain in which it was rapidly rotated to about a max angular velocity of 240 radians per second and decelerated back to 0, all in about 1/100th of a second. And this bottom graph shows a triangular impulse that essentially models that same experimental impulse, but it's much cleaner for the simulation to run. And so this is what I used for my simulations. And then last, I had to assign material properties. And so when assigning these properties, we modeled brain matter, both white and gray, as hyperelastic, which basically just means very stretchy, and nearly incompressible, meaning that any deformations in the matter didn't really affect its total volume. This is reflected by the high bulk modulus, which represents compressibility. These numbers are of order gigapascals, and that's a very high number for bulk modulus. So the higher the number, the more incompressible your material is. And the intraventricular cerebrospinal fluid was modeled as an incompressible liquid. And this is represented by the shear modulus, which shear modulus just looks at stiffness of a material. So liquids have 0 stiffness, and that's why we have a 0 shear modulus here. And as you can see, the gray and white matter also have a pretty low shear modulus, and that's because they are also not very stiff as brain matter. And then here's just the densities of all 3. They're pretty similar, and this is just gotten from experimental data. And so moving on to results, because of the complexity of the brain structure, it was important for me to go step by step in running these simulations. And so here I just start with a simple brain outline with just gray matter. And so obviously not a real brain, but it was a first step. and I applied this rotational impulse. And so here you can see the buildup of stress up to the peak angular velocity at about 5 milliseconds. And from this model, you can see that most of the stress is concentrated around the outer border, which makes sense when you're rotating the brain very fast. And it's of magnitude 10 to the 5th pascals. And this magnitude is typical in brain injuries, so it was a good sign that our simulation was functioning correctly. And so with that, I could add the ventricles to the model, but still excluded any kind of white or gray matter difference. So similar to before, high stress was present on the outer border, but now you can see high areas of stress at the corners of the ventricles compared to other places around the center of the brain. And though the stress around the ventricles didn't seem very prominent, just the fact that we saw some stress at the corners confirmed some of our predictions that there would be more stress concentration if you include the ventricles. And then the final step I was able to reach this summer was to add the white gray matter interface with the white matter slightly varying material properties compared to gray matter. But this is not without, this is without the anisotropy still. So I haven't added any of the axon tracks or directional properties to the white matter. And looking at that simulation, you can see that the stress follows a similar path before, but is slightly disrupted by the white gray matter interface. So even without adding any anisotropy, the white matter outline does have an effect, a slight effect on the force distribution. And so in conclusion, just looking at all three of these results, current results confirm previous studies for the most part. You can see the progression of the models and the fact that there are stress, areas of stress concentrated around the ventricles shows that it's important to model them. And actually going into the future, we believe that including the anisotropy, which is kind of what I did for the first half, was like taking those vectors out of that diffusion tensor imaging experiment, inserting anisotropy into the white matter as part of the simulation will have a very big effect on how stress is distributed, especially on the ventricular region, because it will make the white matter much stiffer in various directions. And so, yeah, that's basically it. I would like to acknowledge Dr. Shenoy and his lab, and Dr. Ayush Khan, who I worked with over the summer, Dr. Jeong and the CEMB, as well as the National Science Foundation for funding my summer. Thank you.
Can I ask a question? Nice job,
Yes.
Noah. I'm wondering if you had discussed it all, or you guys had thought at all about adding the vasculature into the model. I realize you can only do so much in a summer, but I'm wondering ultimately if that might be a useful addition to the white and the gray matter.
I don't think I discussed it this summer, but that is an interesting point that you bring up. And I actually don't have much of a biology background, so I didn't know much about the brain structure coming into this, but that is that's something that I could discuss because I do plan on working on this project past this summer with Dr. Shenoy's lab and Dr. Ayush Khan. So I could definitely talk with him about that. Thank you.
Cool. Yeah.
Yeah. So Becky, that's a good point. But in your mind, are the blood vessels stiffer than the neuronal tracts or softer? That's the primary question.
If I had to guess, I would say they were probably in between the stiffness of white and gray matter.
Okay.
But I don't know.
Yeah,
Yeah.
that's the
Yeah.
primary question. So yeah.
Can I ask a slightly basic question? So when you did your directional analysis thing, how did you determine which data was extraneous in your first model that you did?
Yeah, so the way I did it was looking at, I guess I can go to this slide, looking at the like any individual coordinate point, you can look at the vectors above, below, and to the left and right of it, and you can kind of resolve that like there's only like one vector that is present or one direction that's present all around. And so like if you compare the point that you're looking at to the points around it, you can get rid of any vectors that don't match. And so that's kind of how I did it. And some of these vectors were like also pointing in the wrong direction. So I wasn't only looking at vectors pointing in the same direction, but also like 180 degrees around. And so it helped to look at that. And that's kind of what most of my algorithm did in MATLAB.
Awesome, thanks. Thanks, Noah. That was a great presentation.
Thank you.
So next up, we have Mythili Subbanna from Amherst College. She actually worked with the Anderson Lab at Penn State, and we're lucky to have two students who were able to do their research at Penn State rather than UPenn this summer.
Just checking, can you hear me properly?
Yes.
Okay. So hi, I'm Mythili Subbanna, and I'll be presenting on my project, predicting the turgor pressure-driven mechanical interactions of garden pavement cells during stomatal opening in plants. And this is done in collaboration with Yintong Chen and Drs. Yi, McAvoy, and Anderson. So the system or model that I'm looking at is the stomatal complex in Arabidopsis thaliana. And stomata in general are pores in the plant surface that function in photosynthesis, so how plants make food for themselves, as well as transpiration, so release of water vapor and other gases to the environment. And stomata in Arabidopsis thaliana comprise of two kidney-shaped guard cells that are labeled in green in the image below, and they're surrounded by these larger epidermal or pavement cells that are labeled in pink. These pavement cells can be junctional, so near the top and bottom where the guard cells are touching, or they can also be flanking, so on either side. They can also be both, but for my model, I'll focus on the simple situation where there are two junctional and two flanking pavement cells. So the title is Stomata are dynamic pores, and you might be wondering what makes them dynamic, or how do they open and close? So usually what happens is first there's a stimulus, such as light. So this will be focusing on stomatal opening. When light hits the guard cell, it can activate different photoreceptors, and in general, what happens, among other things in the signaling pathway, is that there's an accumulation of ions in the guard cell as well as the vacuole, so potassium ions and chloride ions. When this happens, there's an increase in the concentration of ions, which means that due to the osmotic pressure gradient, water is more likely to move into the cell because it wants to create equal concentrations inside and outside of the cellular environment. So when water rushes in, that causes the guard cell to swell and turgor pressure to increase, so the pressure inside the guard cell, and eventually that leads to guard cell expansion and the stomata opens. So you might be wondering how the pore actually occurs versus just the guard cells bloating, and that happens in part because guard cells expand anisotropically, which means they don't expand equally in all directions. The cell wall of guard cells contains different polymers, one important one of which is cellulose, which constrains expansion in what you can see as the left and right direction. So the cellulose is radially organized, and this means it promotes, because it's higher in stiffness, it promotes cellular elongation versus expansion just in the XY direction, or just in the X direction, sorry. And so this means it's able to create a better pore for the stomata when it opens, and it's been shown in previous studies that it's actually required for proper stomatal opening. So now moving more into what our model is going to show. Our model is going to explore the interactions between the guard and pavement cells that I mentioned that result from various fluxes in the cellular environment. So this is based off of a previous study that talked about the seesaw hypothesis. So seesaw referring to how the turgor pressure and osmotic pressure sort of go back and forth between stomatal opening and closing. And again, just to reiterate, the guard cells have an influx of ions as well as water, so they expand and they push into the pavement cells, which experience a decrease in turgor pressure during stomatal opening. So what makes this different from previous models is that we'll also be focusing more on the pavement cells. Previous models have either focused solely on the guard cells or solely on pavement cells and how they grow. So we'll be looking at the mechanical interactions between both. We'll obviously be observing the extent of stomatal opening to see how the interactions affect that. And eventually, not shown in this presentation, but we can extend this model to explore changes in cell volume. And via different equations, we can connect this back to turgor pressure and also signaling pathway parameters, such as the rate of pumping and permeability. So putting the model more in a theoretical context as well. So how did we build the model? We used something called the finite element method. By definition, it's a computational method to approximate solutions to system models that may have a complex geometry or defined constraints. More simply, it means that for my experiment, I would create these geometric parts, so the guard cells, as shown in the diagram on the right, and I simulated pavement cells by creating surfaces. So they're not shown here, but they are there, and they're able to exert a pressure on the guard cells. Then I would assign a material that's anisotropic, again, going back to how that allows for the guard cells to expand properly and open the pore. I also assigned an orientation, which is important to account for the cellulose fibers because they're radially organized, as you saw in a previous image. A general rectangular coordinate system isn't ideal, so a spherical system was implemented to account for the curved guard cell. Then I defined some boundary conditions, so when I wanted to stay fixed during the deformation. In this case, this was the top and bottom, so the two junctions, which have been shown in previous experiments to be fixed because of higher stiffness. And then finally, I would define loads, so the turgor pressure within the guard cells and the turgor pressure that the pavement cells are applying on the guard cells. Finally, I ran the simulation, and then I could take different measurements afterwards. Some of the ones I'll show are the complex widths and the pore widths. So the first thing we can note, which corroborates previous studies, is that the anisotropic expansion enables proper pore opening. So again, that's expansion that's unequal in different directions. On the left, you can see the isotropic version, which in the middle, you can see there's barely any pore visible. So really the guard cells are just bloating. They're not really bending in any way. However, when you involve the anisotropy, the pore is much larger, and you can see that overall the guard cells have bent much more. And this is favorable because it accounts for how cellulose functions in the anisotropy by favoring cell elongation and bending. So what we wanted to look at in terms of interactions between the cells is the pavement cell pressurization and how it could constrain stomatal opening. And the way we did this was mirroring experimental results via ablation. So that's using a laser to kill off certain pavement cells. And that was simulated by making the turgor pressure of the pavement cells 0. And I predicted that removing a flanking pavement cells, shown here, would lead to widening of the stomatal pore, and removing a junctional constraint might also lead to minor pore widening, but not so much because we know that it's fairly fixed. So the first thing I did was test out various turgor pressure ratios between guard and pavement cells. And so by turgor pressure ratio, I mean the guard to pavement cell turgor pressure ratio. And so we see that for a larger guard cell pressure, there is a larger percent change in the stomatal complex width. That's almost a linear relationship. But for very small ratios, so less than 1, when the pavement cell pressure is greater than the guard cell turgor pressure, there are no values. And that was because it led to unrealistic stomatal opening. So the pavement cells would expand so much that they would push the guard cells into each other to intersect, which doesn't actually happen in reality. So overall, what we concluded from this slide was that the guard cells need to have a higher turgor pressure to push into the pavement cells and lead to stomatal opening. Next, looking at the ablation results, we concluded that ablating more pavement cells would allow the stomata to open more. Specifically, if you compared when all the pavement cells are ablated to when only a flanking cell is ablated, the pores were able to open wider, 2 times wider in the predicted results and 5 times wider in the experimental. And the reason for this slight difference is that the predicted change in pore width is 2 times larger than the experimental values when only the flanking pavement cell is ablated. So while our results for when all the cells are ablated and junction cells are ablated were accurate, the flanking situation had a slight discrepancy, which I'll go into as a limitation. And overall, just reiterating that ablating the pavement cells allowed greater deformation of the guard cells as showcased by the strain, which allows us to see how much the body has changed compared to its original state. So removal of the pavement cells, which is in this diagram on the right, led to greater expansion and bending and overall a larger pore than when all the pavement cells were present. So overall, what I accomplished was creating a functional stomatal complex model that was surrounded by pavement cells. And we showed that a larger guard cell turgor pressure is necessary to push into the pavement cells and allow for stomatal opening. One limitation was that there were some discrepancies in how guard cell width changes from closed to open state. That isn't accurately predicted, but you can fine tune parameters for that. The implications of this model is that it can be further extended to generate volume results. And as I mentioned earlier, that can be related back to pressure gradients and signaling pathway parameters. So future steps again would be to address the limitations and tune the parameters to better simulate the experimental results, implement the code to generate the volume change results. And our final goal would be to, once generating the volumes, be able to work backwards via equations to predict the turgor pressure of the cells under different treatments, and overall put these in context of the underlying fluxes to understand how the plant cells respond and adapt to their mechanical environment. These are my references. Thank you to everyone who helped, as well as the CEMB for funding my research. That's my presentation.
Great, thank you. If anybody has any questions, please unmute yourself or put it in the chat.
Actually, can I ask a question again? So the ablation of the pavement cells, is that really just, actually just is the wrong word, but is that an experimental technique and obviously a modeling technique just to understand the role of those cells, or are there any actually physiological situations for the plant when that happens? Or is there something different about the way guard cells might develop that are the effective equivalent of that?
So from what I've seen, I mean, laser ablation is a method to kill off the cells, and there are technically different ablation methods. So there's like a chemical version, an optogenetic version. So all of them, I think, are just to simulate when a cell nearby dies and what happens then. So what happens in the absence. So I think it was useful in terms of the absence. But I mean, in terms of the actual method, the laser ablation involves intense light exposure. It is very specific, but I would imagine that I mean, not physiologically, but in an extreme circumstance, intense light exposure could still cause the same effect. And that's what the laser ablation is trying to show. Well, yeah, but in relation to this experiment, we weren't focusing on the intense light exposure. We were more focusing on the presence and absence of the pavement cells.
Yeah, I'm just wondering if, you know, in a plant, when does that happen, if it does happen? I mean, I can see it's an incredibly useful experimental and modeling a technique and assumption, but I just wonder if it actually happens.
Maybe I'll jump in and say that in some plants, it can happen in response to osmotic stresses or drought.
See, okay.
That certain cells will die.
Okay.
Oh, I did have a question, but it looks like we have to move on. And so our next speaker, thank you, Mythili. Our next speaker is also from the Anderson Lab, McHugh Egner from Penn State.
So hi, everyone. My name is McHugh Egner, and I'm in the Anderson Lab at Penn State. And today I will be talking to you about how cell wall patterning in roots is influenced by the mechanical environment in both agar and a hydrogel-based transparent soil. So the overarching question here to my project is, how do roots and cell walls specifically respond to the mechanical environment? Now, little is known about the mechanobiology of plant cell walls and roots. However, most people would agree that roots are essential for plant growth, whether that be the uptake of water, nutrients, or even anchoring down the plant. So understanding how roots and cell walls respond to the mechanical environment more on the cellular level can hopefully one day provide insights for enhancing plant growth and crop yields. So initial experiments involved growing wild-type Arabidopsis plants in a soft and hard media, and it was found that in this softer agar, roots had this circling behavior as visioned in the image on the left, but compared to a harder agar where the roots grew straight down. So next, after seeing these architectural differences, I wanted to dive a little deeper and uncover more of what is changing on the cellular level. So in my project, I am investigating how mechanical strengths of media impact roots on the cellular level, and more specifically to do with cell wall patterning. Now, it is already known that cell wall organization, such as cellulose here, changes as the cell grows and elongates. The fibrils start out in this transverse orientation, and then they reorient themselves as the cell elongates to a more longitudinal formation. However, in this project, we're looking into how other factors like mechanical stimuli also play a role in the cell wall patterning. So we hypothesized that roots grown in a harder media will have different cellulose patterning than roots grown on the surface of or inside a softer media. So to overall test this hypothesis, we grew wild-type Arabidopsis roots in 3 different growth conditions with varying mechanical strengths. The first condition was just growing roots on the surface of slab agar vertically as a control. And then the second condition was growing plant roots actually inside of agar using a soft mechanical strength of 0.4% concentration and a harder mechanical strength of 0.8%. And then the last condition was to grow plants in a near natural media of hydrogel-based transparent soil composed of sodium alginate and phytogel beads, as you can see here in this image. And then once 1/2 ms liquid is added to the beads, it becomes transparent, like here for high resolution imaging. So to continue on with my experiments, I started out by growing wild-type Arabidopsis seedlings in different conditions, as I mentioned before. And then after 6 days, I would remove the seedlings from the media and stain them with ponamine fast scarlet 4B staining, or S4B. And this stain allows for the fluorescence of cellulose and plant cell walls. Next, I conducted confocal microscopy with a laser and took Z-stack images of the cell walls and then analyzed them using a ImageJ software. I used a fibril tool in ImageJ for measuring 2 different things, one being the degree of average fibril orientation in a cell wall, where I was able to measure the orientation of each slice in a Z-stack and then compile them all together. And then the second was the average anisotropy of these fibril arrays, or in other words, how well the cellulose fibrils were ordered. So to begin with, one of the main results that I found was that roots grown on the surface of agar had cell walls with more of a variety of cellulose orientations and a lower anisotropy than roots grown inside of agar. So in the image on the left, you can see that these lines indicate different orientations of each slice or level of the cell wall. And then once they were all compiled on top of each other to a maximum projection, you can see how the average orientations vary throughout the cell wall and aren't quite exactly aligned. Now then, comparing that to the right image here of roots that were grown inside of agar, the lines are they're more aligned together and actually longer in length, indicating a higher anisotropy. Now, one limitation is that it can be hard to compare exactly one cell to another, as they might be in different stages of elongation or growth. But I collected many samples of cells all within the elongation zone of roots and the root tip to average out my results for each of the different growth conditions. So here shows a collective diagram of quantitative data regarding the variability of the cellulose orientation within each condition, kind of from the images in the last slide. And it was found that roots grown on the surface of agar had the highest average variability in orientation right here than roots grown in other conditions. Another finding is that roots grown inside of transparent soil had a higher variability of orientation than inside of the agar growth condition. And lastly, when comparing different mechanical strengths of agar right here, the roots grown in the 0.8% agar had a higher average variability than those grown inside of 0.4% agar. So to move on, this here sums up each condition in regards to the second measurement I conducted involving average anisotropy of the cellulose arrays. So in here we found that roots grown on the surface of agar had a lower anisotropy than those grown inside of agar, and inside of 0.8% transparent soil. We also found that when comparing mechanical strengths again of the agar, that roots grown inside of the 0.8% agar had less anisotropy than those grown inside of the 0.4%. And the last key takeaway is that roots grown in the 0.7% transparent soil had the lowest anisotropy than all other conditions, including the roots that were grown on the surface. So overall, the results that we found support our initial hypothesis that there is a difference in the cell wall patternings when comparing roots grown on the surface of and inside media, and also within varying mechanical strengths in media. So to kind of sum up some of these results when comparing the roots grown on the surface of agar to inside of media, surface grown roots had cellulose orientations that varied more across the layers of the cell wall, meaning that they weren't all aligned together and in the same direction, compared to orientations found in roots grown inside of media, such as the agar, where the cellulose fibrils were more oriented in the same direction, overall indicating that mechanical stimuli of media in the roots of roots grown inside of media influence the cell wall patterning in some way. Now, possible reasoning behind this difference is it might be harder for roots to push through a media like a solid slab agar. And in order for a cell to overcome this and continue to elongate inside of the media, fibrils might have to align more together. And also then when comparing different growth conditions, roots grown inside of the transparent soil beads varied more in their cellulose orientations than those grown inside of agar. Now, to possibly account for this difference might be due to the differences in mechanical properties of these two different growth medias. So agar is more of a solid gel that compresses the root fully all the way around it, while transparent soil, as mentioned earlier, is composed of beads. And when beads are stacked on top of each other, there still is some slight gaps in between them or air pockets to give a more natural-like state. So lastly, when looking into the differences between mechanical strengths or a hard and softer agar, roots grown in the harder agar varied more in the cellulose orientation than those grown in softer agar. So this can suggest that the different mechanical strengths of a media influence the behavior of cell wall patterning as well. So overall, some future directions include studying the mechanisms behind these differences found in the cell wall patterning and when they're influenced by the mechanical environment, and also attempting to conduct high resolution imaging of cell walls without actually having to remove the seedlings from the growth media for staining purposes. So on the right here is a cover slip chamber slide with transparent soil inside and attempting to grow roots in this chamber slide. And if the roots are close enough to the cover slip, focusing with a confocal microscope would be possible, and also staining within this system. I would like to thank the CEMB along with Annie and my lab and Dr. Anderson. Are there any questions?
I have a question. Thinking about the transparent soil, I guess, do you see like micro level effects? So like parts of the root that are touching the beads versus the ones that are in the spaces, or is it more of like a root level effect?
So yeah, that is something I'm trying to look into, because once you actually add the 1/2 ms into the transparent soil to make it transparent, you can't see the beads anymore. So it's hard to tell now. Possible things to look forward to that I've talked to Dr. Anderson was looking for some type of way to fluoresce the beads so you can see where it's going and the difference of where the root is compared to where the bead is. So it's a great question. Thank you.
Are these beads like the toy ones that inflate, the polymers that kind of inflate?
They're
Because they sell in lots of different colors. I'm just curious.
yeah, I don't know if like kind of like the size of like the little Orbeez beads, but and you can vary the size as well. But yeah.
I have a question. The difference in the concentrations of the agar are interpreted as due to the stiffness of the agar?
So yeah, the 0.4% concentration would he suggested as a softer media, and then
And is it known what the limit of force is that the Arabidopsis root can generate? Is it anywhere near like a stalling force in one of these agar materials?
Stalling force, you mean like it wouldn't be able to grow inside?
Yeah, yeah. I mean, it's nowhere near like running into a rock or anything.
No, so sometimes in the 0.8% agar, sometimes the roots aren't able to penetrate, but some do grow in. But no, it wouldn't be as much as a rock. I'm not exactly sure what.
It's interesting. So if they can't penetrate, do you know why they can't penetrate?
Not quite. Not necessarily, I wouldn't, but something to look into.
Thanks.
I'll just add one
Awesome,
thing,
great
which is that there are very cool experiments where people have done interfaces between low and high percent agar, much higher than a 0.8% agar, and they see the roots actually grow down and grow along the surface, but find an area of weakness to be able to grow.
Awesome, great. Thank you, Matthew.
Thank you.
So our next speaker is Kaitlyn McHugh. She will be presenting her work in the Osuji lab.
Hi guys, I'm Kaitlyn, and as Dr. John said, I've been working with the Osuji lab all summer, and my project focused on the synthesis, characterization, and flow behavior of different biomaterial-based microfibers. So first, I just want to talk about what a microfiber is and why they're important. So these materials are on the order of microns in diameter, and they're really interesting because you can control different properties of them, like their length, diameter, mechanical strength, elasticity, based on how you make them. And they've become an important tool in a bunch of medical applications. For example, they've been used as scaffolds, as shown here on the bottom left, in tissue engineering, so you can actually attach cells and they can move along these fibers. And then they've also been used a lot in drug delivery. Based on the composition of the fibers, you can actually modify them and attach your drugs to the surface of them. And you can also encase your drug into those hollow fibers as well. So despite being used in a lot of these medical applications, they're often made using synthetic polymers like polyacrylonitrile just because they have really good mechanical properties. However, they're often not as biocompatible or degradable as natural materials. That's why we decided to look into natural alginate and nanocellulose fibers and compare them to pan fibers in respect to their synthesis, characterization, and flow behavior. So I'm going to talk about the materials we used. As I said, for the synthetic material, we went with polyacrylonitrile, and this is a fossil fuel-based polymer, so it's pretty detrimental to the environment to produce it. However, it does have really good mechanical properties. And in terms of natural materials, we worked with cellulose nanofibers, which come from refining plant cells down to individual fibers. And they're really small in diameter as well as length, and also biocompatible. And the other natural material we worked with was sodium alginate, and it's pretty similar in structure to the cellulose, and it comes from brown algae. It's also non-toxic and pretty low in cost. And all these materials have some advantages and disadvantages, but there is definitely a need for a material that has both good mechanical properties as well as good biocompatibility. So just an outline of what we did for this project. First, we had to make our precursor to the fibers, and then we used that precursor in the wet spinning process to make the actual fibers. And once we had that down, we sort of changed up the wet spinning process to control different dimensions of the fibers, such as diameter as well as length. And then we used those fibers for our characterization steps using an optical microscope as well as a rheometer. So starting with the precursor synthesis, it's pretty simple. It's basically just dissolving a polymer into a solvent. So we made individual solutions of the pan, sodium alginate, and CNF, but we also did some mixtures of the sodium alginate with the CNF. And the pan we dissolved in DMF, while the sodium alginate and CNF mixtures were dissolved in water or suspended in water. So then we used those precursors for wet spinning. And in this process, the polymer dissolved in a solvent is just going to be pushed through some tubing and through a needle into a coagulation bath of non-solvent to form microfibers. And for the polyacrylonitrile, the PAN fibers, it's just going to be a precipitation reaction in which the PAN is dissolved in DMF. However, it is not soluble in water. So when we mix this PAN and DMF with the water through the extrusion needle, the PAN will precipitate to form fibers. But for the sodium alginate and CNF, it's slightly more complicated. So the sodium alginate is soluble in water, but calcium alginate is not. So when we extrude the sodium alginate and water into a calcium chloride coagulation bath, an ion exchange reaction occurs between the sodium and the calcium, and you're actually going to have calcium alginate precipitating to form fibers. So after we continuously wet spun our fibers, we decided to change up some of the dimensions, starting with the diameter. So we found that we could control the diameter by flowing the precursor at different rates through the extrusion needle. And we measured this under the optical microscope. Some images of those are shown here for the smallest fibers we obtained for the PAN, the 2% sodium alginate, and the 2% SA and 0.5% CNF. And we found an interesting pattern that the precursors followed. That was a slower rate would make a smaller diameter, which is kind of the opposite of what we predicted. And I just have the size distribution shown here. And another trend that we found was that PAN actually needed a slower rate to yield the target fiber size of small diameters. And the smallest we got was actually around 100 microns. But for alginate, we could pump at even faster rates and get even smaller diameters, with the averages being 38 and 87 microns. And this may have to do with how the precursors are exiting the needle or the viscosity of our precursors. So we also decided to look at the length of the fibers, and we controlled this by cutting the fibers with air during extrusion. So we used two different syringe pumps so we could pump the air and precursor at different rates. And basically the air would intersect the flow of the precursor to create a pattern flow of precursor and air, so it would make separate fibers. And I just have the size distributions again shown on the bottom for length for all of the sodium alginate and sodium alginate CNF mixtures. We found that adding CNF resulted in the fibers becoming shorter compared to the sodium alginate on its own. And this may be due to the structure of the sodium alginate fibers, that CNF may interrupt that final structure. And then onto the characterization steps, we were able to characterize our precursors by their viscosity using a rheometer, which just studies how a solution flows. So we use this to track the stress as a function of shear rate. And it was pretty simple for the PAN and sodium alginate precursors. They behaved as Newtonian fluids, which had constant viscosity, so it's just a straight line. However, it got more interesting with the CNF fibers. They showed non-Newtonian behavior, meaning that the viscosity would change as the shear rate increases. And this has interesting effects for how the fibers are extruding through the needle and basically the overall morphology of the final fibers. We also characterized the actual fibers through tensile measurements, and we were able to determine the Young's modulus of some of them using a different configuration on the rheometer. So basically, we just would stretch the fibers until they breaked, and this instrument would allow us to track stress as a function of strain. And this tells us basically just how easily a material stretches. So this graph here shows the stress as a function of strain, and the slope will give us the Young's modulus. So we found that the 2% sodium alginate and 0.5% CNF actually had the highest Young's modulus. And then in second was just the 2% sodium alginate. And this is a lot higher than the fibers containing the 1% sodium alginate, meaning that these fibers were more elastic. And then for the PAN, it's shown in purple, and it's very short, meaning that they are a lot more stiff and they break at a lot lower strains compared to the 2% sodium alginate fibers. So to conclude, from the study, we found that a lot of different factors can influence the size, structure, and mechanical properties of our fibers, such as extrusion rate, needle size, precursor composition, and viscosity. But we were able to make these fibers, these sodium alginate and cellulose fibers, by wet spinning, and they were of comparable diameter to PAN. And then we were able to characterize these fibers, finding that the PAN fibers were more stiff, while the higher percent alginate fibers were more elastic. And then we found that differences in things like viscosity and extrusion rate can impact the final fiber structure. And just looking into what we can do with this in the future, some interesting directions to look into are the biocompatibility and degradation of natural and synthetic fibers for use in medicine. For example, if you want something to be biocompatible, you're going to want the degradation products to also be non-toxic. And for a perspective from tissue engineering, you want the degradation rate of your fibers to be similar to your target cell or target tissue. And lastly, I just want to thank everyone who helped me with this project, my mentor Ning, Dr. Osuji, and the whole Osuji group for all their help this summer. And if anyone has any questions, let me know.
Great, thank you, Kaitlyn. If anybody has any questions, please feel free to unmute yourself or put it in the chat.
This is Paul Janmey. How do the fibers interact with each other? Are they sticky to each other?
Yeah, that's a good question. So it was actually interesting. So the polyacryl and nitrile fibers didn't really stick to each other, but the alginate and CNF fibers were a lot more sticky, like they would get very entangled.
Oh. Oh. So you think they stick by the sides, not because they've branched during the polymerization process?
Yeah, because you can separate them into like a single continuous fiber.
Oh, that's really interesting.
All right, if there's no other questions, thanks, Kaitlyn. Our next speaker is Elizabeth Seidl from Kansas State University. She'll be talking about the work that she did in the Discher lab.
Hello, everyone. My name is Elizabeth Seidl, and I'm going to be talking to you guys today about actomycin's and oxidative stress's effect on the cell genome expression of a heart and the heart beating pattern. Okay, so first, just a little bit of background on DNA damage's effect on cells. So DNA damage happens when DNA repair factors become mislocalized within a cell, which causes the nucleus to rupture. So this causes an accumulation of DNA damage, which can then lead to premature aging due to the telomere shortening and irregular beating within the heart. Okay, so more background. It's important to understand actomycin and collagen's role in the cells. So this might be a little bit of review, but actomycin is in the cytoskeleton and essentially controls the main complex of beating of the heart. And so there's a normal level of actomycin stress present when the heart is beating normally. Collagen also is present in the ECM, and it is a major factor in the tissue stiffness. And I just put a picture here of an embryonic chicken heart. So we work with a day 5 embryonic chicken heart. Okay, so moving on. The experimental treatments that we use are blebbistatin, which is a drug which is a myosin inhibitor. So this actually inhibits actomycin stress. And then we also use H2O2, which is going to cause the oxidative stress, and that is a known control for DNA damage. And then our DMSO samples are just going to act as our control. Okay, so we have 3 main objectives for my summer project. So the first was to look at the genomic expression of DNA damage within the hearts due to both the actomycin and the oxidative stress. The second is to look at the beating patterns of the hearts when we use this blebbistatin drug. And then finally, we want to look at characterizing the collagen and myosin when using blebbistatin. So we're going to look at doing these 3 objectives in 3 different ways and 3 different parts of my project. Okay, so the first way, so we're looking at the gene expression levels. So we're going to use single cell sequencing cluster maps to analyze the expression levels. Then we're going to look at the frequency of the beating, and we're going to do that using image J analysis. And then to characterize the collagen and the myosin, we're going to use second harmonic generation imaging. So to start, I'm going to talk about the first part of the three, which is the single cell sequencing. Okay, so it's important to understand, once again, these 4 experimental groups. So blevestatin actually stops the beating of the heart. So we have a second group, which is the blevestatin with washout. So when we wash out the blevestatin with media, the heart is able to regain beating. So we take these 4 experimental groups, and then they go through single cell sequencing. Just a brief, if you don't know what that is, we take all the cells and attach a barcode to them. So we have their genomic expression, and we put that in a large matrix that we're able to analyze. I analyze that using specifically the Seurat package within R. Okay, so using that package, I'm able to do dimensional analysis on it to create a UMAP. What this UMAP shows is it clusters based on genomic similarities. So in the different colors in the labels, you can see that I've identified cell types that are present within our sample. So these are all present within the embryonic heart. And so I did this using significant biomarkers that Seurat was able to identify for me during the clusters. And then I used literature and constructed this table for these are the biomarkers I've used throughout the summer to identify each cell type. So then using that, we're able to move forward by doing an actual gene expression test. So doing gene expression tests, we use the standard Wilcox test. And I found that these 2 genes, these are both mitotic genes, were downregulated within the sample that was treated with blevestatin and then washed out to restart the heart and the oxidative stress. So these are significantly downregulated. So this can tell us that there's possibly a link between oxidative stress and the myosin inhibition with the washout that could cause cell cycle arrest. Okay, so now we're moving to the second part of the project, which is looking at the effect of the frequency on the heart. Okay, so we have 3 different experimental groups. So we have our controls to start, and then we have the hearts that are treated with blevestatin and allowed to continue all the way through before washout. And then we have hearts that are washed out early at 45 minutes and then allowed to restart beating. Okay, so to analyze the heart beating, I have a delta AR, which is the change in aspect ratio. So that takes the X and Y axis of an ellipse used to fit the heart. And then we're able to analyze that to find the heart beating pattern. Then we use the discrete Fourier transform to actually look at the prominent frequencies within the heart beating pattern. So you can see a shift here when we have our T equals 0 minutes, and then in the red, we have our T equals 90 minutes. So we can see a change in the dominant frequency. We also look at the amplitude, which is the average beating strain, by taking the average of the peaks minus the average of the valleys. So our data look like this. And so we have our 3 different experimental groups. So in the gray shaded areas is where we treated it with blevestatin. So as you can see in that 3rd column, that's why we only have the gray shaded area till 45 minutes, because that's when it was washed out. So we can see some significant changes within the frequency when the blevestatin is applied. We also see a big change in the actual beating pattern of the heart. So we can conclude that the myosin inhibition does affect the beating pattern and the frequency. Okay, so now my 3rd and final part of my project, to characterize the collagen myosin levels. So our SHG imaging, if you're not familiar, we shine a laser through the samples, both in the backward and forward direction. So we looked at it at different intensities and different wavelengths. We then concluded to focus on the forward. As you can see below, the backward direction has a lot of noise. So that's above, we concluded to look at 920 nanometers based on literature. We did this by identifying 3 areas of the heart and then 2 backgrounds. Then, as you can see on the far right, we looked at where the intensity peaked. So as we move deeper into the heart, we can see an increase in an eventual peak in the intensity. We then averaged the 7 points around the peak. So we were able to average intensity for one region of the heart. Okay, so my results with this is we were actually able to see some heterogeneity within the individual hearts. and within the different regions of the heart. So for each heart, we analyzed a ventricle and a non-ventricle, and we can see some significant differences, which tells us that there's a significant difference not only within the blebbistatin and the control hearts of the collagen and myosin expression. Okay, so to conclude the 3 parts of the project, we saw a possible link between oxidative and myosin inhibition with washout to cell cycle arrest based on those mitotic genes being downregulated. We also saw the myosin inhibition affected the beating pattern and the frequency of the hearts. And then we were able to see some heterogeneity concerning the collagen and myosin levels from the FHG imaging within the heart samples. So as far as future work, we kind of have future work for each of the 3 areas of the project. So we want to then take that gene differential expression test onto those UMaps with those identified cell types that I worked on, and look at how the gene expression is different within the cell types. We also want to look at how etoposide and other DNA damage drugs affect the beating of the heart. And then finally, for the FHG imaging, we want to actually look at the differences between the collagen and the myosin respectively within those intensity levels. So there's a lot of work still to be done, but I'm really happy with the work we've achieved this summer. So finally, I just want to thank my mentors, Mística, Karan, Mai, and Manu, and especially Dr. Discher for letting me be in the lab this summer. It was a great experience, and I'd be happy to answer any questions.
Hi, can I ask a question, Elizabeth? So a very nice presentation and nice work. So were you surprised at all by the differences based on location? In other words, the ventricular and non-ventricular areas. Did that come as a surprise, and what do you make of that?
Yeah, so that personally came as a little bit of a surprise to me. We were also surprised that we saw a difference within the blebbistatin areas as well as the controlled. I think we expected to see it within the control, but the fact that it was expressed differently within the blebbistatin. I don't have a great reasoning why at this point. This is kind of data that we've just found. But I think it's definitely interesting steps going forward as we look to compare within the different treatments. We haven't seen a big like significant difference between the different treatments, just within the different regions at this point.
Thanks.
Great, thank you, Elizabeth. So our next speaker will be Bruce Enzmann. He's a student at Johns Hopkins University, and he did his second summer with us actually in the Burdick lab.
Yeah, hi, thank you so much for having me, and thank you all for coming again. For the past month, I've had the pleasure of working with John Galarraga, Abhishek Dand, Nico DiCaprio, Dr. Claire Witherell, and the Burdick lab at Penn to model hydrolytically degradable thiolene cross-linked hyaluronic acid hydrogels. So hydrogels are water-swollen polymer networks that show promise for a range of biomedical applications in recent years, such as tissue engineering and disease modeling, serving as vectors for drug delivery and providing materials for techniques in biofabrication. Hydrogels are particularly useful for these applications since they may encapsulate therapeutics or cells, they exhibit biocompatibility, they are porous, so they can permit transport of macromolecules throughout the network, and they are often biodegradable. In particular, hydrogel systems that are degradable have previously been shown to modulate cellular mechanosensing and behaviors. And in one instance, our lab has previously used an enzymatically degradable hydrogel system, which can be seen here breaking the crosslinks in the presence of proteases to investigate the effects of cell-mediated nascent matrix formation. Alternatively, a previous study from Dr. Wells and Dr. Burdick's labs has used a hydrolytically degradable hydrogel system, shown here breaking crosslinks as a consequence of hydrolysis, to investigate the effects of fibrosis regression on hepatic stellate cell actin cytoskeleton reorganization. Yet on a broader scale, a proteolytically degradable system represents the capability for cell-mediated degradation isolated from materials design, whereas hydrolytically degradable systems enable user-defined degradation for tissue engineering applications. So these studies highlight an imperative need that in order to improve our control over cellular mechanosensing in hydrogels, degradable hydrogels with easily tunable mechanics and degradation times are needed. And for us, we particularly wanted to focus on those that undergo hydrolytic degradation. In some instances, hydrolytically degradable gels degrade on relatively faster timescales, but for some applications, we need gels that degrade on longer timescales. Thus, we also need models that can predict and screen for hydrogel formulations with target mechanics and degradation profiles. So to design these degradable hydrogels with tunable mechanics and degradation times, our lab has been excited about this developed chemistry using sodium hyaluronic acid and carbic anhydride to fabricate norbornene modified hyaluronic acid with a norbornene group shown up here in the green. This also contains hydrolytically degradable ester bonds, which are now shown here in this blue box. And we can then combine NORHA-CA with a diethyl crosslinker like DTT and a photoinitiator to crosslink into a hydrolytically degradable hydrogel network via thiolene crosslinking with visible light. And this polymer system circumvents some previous limitations in hydrolytically degradable hydrogels by providing multiple modalities with which to tune mechanical properties and degradation times, as I mentioned before. So to examine this modularity of our hydrogel system, we fabricated NORHA-CA at 14% degree of modification, 30% degree of modification, and 40% degree of modification shown here, and then examined their resulting storage moduli. You can see that all formulations quickly cross-linked after application of visible light, which is shown by this sharp increase in storage moduli. And within each modification group, we controlled the final concentration of functionalized polymer before gelation to further change this storage moduli. We also demonstrated that we were able to readily tune the compressive moduli of hydrogels from our polymer system, again, via changes in degree of modification, as well as by changing this functionalized polymer concentration. So thus far, we have shown that we can tune the mechanics of our polymer system through multiple modalities. But next, we also wanted to show if we could just as easily tune the degradation of these hydrogel networks. And to do so, we again made hydrogels using our hydrolytically degradable NORHA-CA gels with varying degrees of modification, polymer concentration, and cross-linking efficiency. We then placed these hydrogels in separate 1.5 milliliter Eppendorf tubes that were filled with about 400 microliters of cured PDMS, shown here in red, and a milliliter of phosphate buffered saline. And then these tubes were incubated at 37 degrees C on a platform rocker to model free swelling conditions. And at a given experimental time point, we would then collect the supernatant from our degradation and replace the 1 milliliter of PBS before returning to the incubator. Here, the cured PDMS, shown here in red, was used to make this supernatant collection a lot easier. And then at some final time point where the samples were determined to be degraded, we fully degraded all of the remaining HA in the tubes. And lastly, we used a previously reported uronic acid assay to analyze these collected samples for degraded HA concentration over time. And here you can see our 14% modification NORHA-CA fabricated at 3 weight percent and 50% cross-linking, which degrades on the scale of 8 days. Adding on to this, you can see our 14% NORHA-CA at 5 weight percent and 50% cross-linking, which degrades slightly longer on the scale of 9 days. And then the 40% modification NORHA-CA gels that were made at 3 weight percent and 100% cross-linking already demonstrate these longer degradation times at about 11 days. And then lastly, the 40% modification NORHA-CA gels at 5 weight percent and 100% cross-linking had the longest degradation times at about 14 days. And these representative degradation profiles of the 14% and 40% modifications further demonstrate the range of degradation times we were able to obtain. So thus, alongside these tunable mechanics that I've shown, we found that hydrogel formulation can modulate this degradation timescale, again, indicating the ability to tune degradation time using multiple different avenues. So after experimentally collecting degradation profiles and demonstrating the tunable degradability of our hydrogels, we wanted to develop a MATLAB model that could capture the degradation properties of these hydrogels. And in doing so, from gelation theory, we were able to determine a required fraction of intact crosslink nodes that are needed to maintain a hydrogel network, and this is called P disintegration. And we did this for each experimental hydrogel. And previous literature also suggests that hydrolytic degradation can be assumed to follow pseudo first order kinetics shown here by this rate equation, which then down the line allows prediction of hydrogel network disintegration times. But still, in order to predict disintegration times for these experimental hydrogels, we needed to interpolate these pseudo first order kinetic degradation constants from the degradation profiles that I just previously showed. So it is important to note that interpolating these K values from the degradation profiles, we applied an exponential fit to each gel individually with n equals 3 gels per weight percent. But then we excluded these last day time points where we artificially determine the degradation time during collections to get the fits to be more accurate. And here you can see degradation constants for the 14% modified NOR-HCA at 3 weight percent and 50% cross-linking, which gave us a predicted disintegration time of 9.8 days, as well as 14% modified NOR-HCA at 5 weight percent and 50% cross-linking, which gave us a degradation time of 10.4 days. We also did the same thing for the 40% modified gels at 3 weight percent and 100% cross-linking, giving us a disintegration time of 13.8 days. And again, the 5 weight percent with 100% cross-linking, giving us the longest predicted disintegration time of 19.1 days. So after using gelation theory to predict the disintegration times of our hydrogel networks, we further implemented a Monte Carlo stochastic modeling technique to the MATLAB code, wherein each of these green representative cross-linking nodes is assigned an arbitrary lifetime that is related to those previously interpolated K values. And after some time, you can imagine that this representative network might start having cross-link nodes degrade, causing some of these chains to break off from the network, which we wanted the model to capture via this predicted degradation profile. After some longer time, you can imagine that an even larger number of these cross-link nodes will start degrading, causing many more of these polymer chains to break off. And then finally, at some point in time, the chains of the network will completely break off, which represents more of what occurs in a fully degraded hydrogel system. Thus, we were able to use our stochastic MATLAB model to develop predicted degradation profiles for the 14% modification NOR-HCA at 3 and 5 weight percents, in addition to the 40% modification NOR-HCA at 3 and 5 weight percents, using those previously interpolated K values from the experimental degradation profiles. And again, these profiles demonstrate the range of degradation times that we were able to obtain, yet these profiles still demonstrate some of this relatively fast degradation. So we expect that with further development, we will be able to predict degradation behavior of mixed polymer hydrogel networks using these results in order to target longer degradation times with relevant mechanical properties. So overall, we began synthesizing hydrolytically degradable hydrogel networks via norbornene modified hyaluronic acid thiolene cross-linking with visible light. We then show that this hydrogel system exhibits tunable storage and compressive moduli via degree of modification and polymer concentration. And then lastly, we used a previously reported uronic acid assay to obtain degradation profiles of our hydrogels, which we then incorporated into a MATLAB model to predict disintegration times and degradation profiles for each experimental gel. So I want to take this time to thank the Burdick Lab and especially my mentor, John Galarraga, Abhishek Don, Nico DiCaprio, Dr. Claire Witherall, and Dr. Jason Burdick for letting me work with them this summer. I also want to thank Dr. Annie Jeong for doing a fantastic job advising us in the program this summer and last summer. And lastly, I want to thank the CEMB and NSF for hosting us and funding us for the summer. Thank you.
If there are any questions, please feel free to unmute yourself and ask. I have a question. If you go back just to your last slide, so your model prediction and your observed days are a little bit off. Is that not significant? Because it does look like they're also like with each one of those kind of the model and the observed are kind of differing more and more. Can you explain that a little more?
Yeah, so the differences that we're seeing here are largely due to the experimental setup. So you can imagine that in collecting the supernatant from these gels every day, we're kind of disturbing the gels, we're messing around with them. But also, and then at the same time, when we can no longer pipette the supernatant out of these Eppendorf tubes, that's kind of when we're determining artificially that these gels have degraded. So we do expect, without disturbing them anyways, that these degradation times would be longer. But again, there are some limitations in this model wherein we're not really accounting for non-idealities in degrading hydrogel networks, such as chain entanglement and small molecule diffusion. So we're hoping that this is something we can iterate in the future.
I have a question. Can you hear me?
Yes.
Hello? Yeah. So are you using rigidity percolation here at all? Or is it sort of solubilization? It sounded like these measurements you make are based on release rather than mechanical measurements for percolation, gelation, which would be a rigidity percolation.
So using the uronic acid assay study, we're essentially looking at release rate concentration over time for these hydrogels. So we're not doing anything with the mechanical properties degrading over time.
So not really what we would say is gelation. Yeah. Okay. Thanks.
Awesome. Great job, Bruce. Thank you. So our next speaker is Nathan Rojas Ocampo from Columbia University. He'll be presenting on the work he did in the Discher lab.
Does it show up well? All right, thank you. All right. Good afternoon. My name is Nathan Ocampo. I'm a rising junior studying biomedical engineering at Columbia University. This summer I worked in the Discher lab where my project was focused on identifying cell types and developing hearts, as well as measuring the levels of collagen and myosin using novel methods. Our project was inspired by a 2019 paper published from the Discher lab that revealed the mechanosensitivity of lamin A. This paper revealed the mechanism through which lamin A protects the cell from DNA damage in response to ECM stiffness. So when the ECM gets stiffer, it causes stress in the cytoskeleton, specifically actomyosin, and this actomyosin is connected to the nucleus, and as the tension increases, it can lead to nuclear rupture. This nuclear rupture can lead to loss of repair factors, which leads to detrimental DNA damage and telomere attrition, which could lead to premature aging. In order to study this relationship further, we wanted to use embryonic chick hearts to look more into DNA damage and actomyosin. So why did we use embryonic chicken hearts? Well, the chicken hearts, the beating in the hearts causes actomyosin tension, so it's a very easy way to access that. But we're also able to see physiological effects with different experimental treatments. Through adding these different treatments, we're able to see things such as DNA damage. We're able to identify different cell types and look at the levels of collagen and myosin. So for our experimental setup, there were two major components of the project. The first part, we'd have the single cell RNA sequencing. And the second part over here, we'd have an ex vivo heart experiments where we'd kind of look at the heartbeat, which would give us good indication of heart function, as well as second harmonic microscopy, which would give us more information about heart composition. So for right now, we're just going to focus on the single cell RNA sequencing part. So here we have a representation of the different treatments. This is a figure that Elizabeth made. And here we have the control, blebistatin washout, and hydrogen peroxide. So the blebistatin is a myosin inhibitor. And inside the heart cells, heart muscle cells, the myosin is what leads to contractions. So inhibiting this myosin, we could lead to smaller contractions. And that's what we would see in this treatment. With the blood washout, we wanted to see whether or not the heart was able to recover. So we would add the blebbistatin, and then at a certain time point, we'd wash out the media, and then we'd see whether the contractions were able to come back. So single cell RNA sequencing gave us gene expression at a cellular level. And the way it does this is that we take a tissue, we break it down into its constitutive cells, and then we take each cell, look at the gene expression for each cell, and then we gather it all in this matrix. And then we do that for all the cells that we process through the single cell RNA-seq. From there, we're able to represent it visually using a heat map, or over here where we use UMaps. And that's what we ended up doing. So we used principal component analysis to reduce the dimensionality. You get a lot of like genes and data points for each cell. And then we'd cluster them together with UMAP algorithm. And in that way, we'd be able to look at the different cell types. So now that we've covered the single cell RNA-seq, we could move into the ex vivo heart experiments. Let's talk about that. So the experimental setup was basically the same. We had the control, the bleb washout, except this time we didn't have the hydrogen peroxide. We just stuck with these three conditions. So the first thing that we were dealing with was looking at heart function. And in the top, you can see a little movie of the heart beating. And what we worked to do was quantify this heart beating. And we did that using the aspect ratio of the heart. Through the aspect ratio, we could see the beating strain, which related to the contraction size. And then we'd also see how often it would beat, which would give us the beating frequency. So those are some of the parameters that we would use to gauge heart function. With the second image here, this is our second harmonic microscopy. So right here, we're kind of moving into the heart. This is the heart tissue here, and then over here is some background signal. What we would do is select certain regions of the heart, and then we'd calculate the intensity of the signal in order to make conclusions about the protein makeup. So this is some of the workflow that we would do for the beating pattern of the heart. We would graph the changes in aspect ratio, and then from there we'd filter out small fluctuations so we could work with cleaner data. And with this, we were able to calculate the average beating strain. So we were able to do discrete Fourier transform to find frequencies that occurred with the hearts. So we would be able to identify the frequency which the heart was beating at different time points throughout the experiment. For the SHG data, we gathered different ROIs. So we take these three and then subtract away the background. And this would give us a characteristic curve that we would measure with depth. And from there, we would take a ranged average of this peak value. And this gives us a characteristic point that would allow us to compare between different regions of the heart. So all of this manipulation and quality control was extremely time consuming. So I was able to develop a Python script that could record the heart beating data as well as the SHD data. And then it was able to organize the raw data and turn it into Excel plots, which reduced the time from days for a file to minutes for pretty much all of them. And then the final thing was it was uploaded to GitHub to allow for collaboration during the project, but also for future use in the lab. So results and discussions, back to the single cell RNA-seq part. My part of the project focused on identifying cell markers. So Elizabeth and the other REU students we were working with could make conclusions about DNA damage and mechanosensitive proteins. Through literature surveys, I was able to identify hemoglobin proteins that could identify red blood cells, troponin that would identify cardiomyocytes, and transcription factors that would identify fibroblasts. In terms of the heart, the ex vivo heart experiments, we're able to see significant change when treated with blebbistatin. So this slide is a breakdown of the last slide. In the top, we can see how the controls stayed the same throughout the experiment. However, bleb and bleb washout had significant increases from from the time zero when they were added, the treatments were added. We also see how the strain or the contraction size of the control stayed the same throughout the experiment. However, bleb and bleb washout tended to decrease, and even though there was a recovery in the bleb washout. So now when we look at the SHG, so this is kind of looking at the heart composition, and so we're able to see how the forward, the bleb washout and the bleb didn't really have any differences in SHG signal. However, the control did, and this shows heterogeneity between the ventricles and the non-ventricles, and it could be related to higher collagen content in the non-ventricles because it produces a higher SHG signal. So concluding, our compilation of the cell markers allowed for cell types to be identified. The heart beating patterns were sensitive to the inhibition of myosin. And then the SHG allowed us to identify the differences in heart composition in versus in the non-ventricle and ventricle regions. Our future work includes pairing phase contrasting, so phase contrast imaging, which is the heart beating videos with the SHG, along with the single cell RNA-seq data of the hydrogen peroxide treated hearts to reveal the effect of DNA damage on the heart function as well as composition. And then another thing that I'm excited to work on is this heart image segmentation. So this fall, I'll be continuing working on segmenting the heart into non-ventricle and ventricle regions. Doing so, I'll be able to pair the SHG data with beating data for specific regions of the heart, which will allow us to conclude more accurately with the different regions. So sometimes there's more strain here than there is here, and doing so will allow us to identify that. And I just wanted to acknowledge Karan, Manasvita, Mai, Manu, and Dr. Discher for having me in the lab and helping me with this project. I wanted to thank Elizabeth and the other RU students who work together. And I want to thank Annie for putting together this program. Thank you.
Great. Thanks, Nathan. I think we can take one question. Maybe you're off the hook, Nathan. Thanks again. Let's move on to our next speaker. Our next speaker is Katherine Kerr from Purdue University. She will be talking about the work she did in the Janmey lab, and I believe this is her second summer with us as well.
Hi, Annie. Thank you so much for the introduction. As Annie said, my name is Katherine Kerr. This summer, I was a member of the Janmey lab, and today I'll be giving my final presentation, which is an extension of my project from last year on the viscoelastic properties of tofu as a phantom for liver disease diagnosis. So as many of you probably know, liver disease is a major issue worldwide. It's the cause of approximately 2 million deaths annually. And on the right, you can see a healthy liver, and now you see a fibrotic liver. So many liver diseases, such as liver fibrosis, shown here, are associated with mechanical properties, changes in the mechanical properties of the tissue, including increases in stiffness. In fact, liver stiffness can predict incidence rate of hepatocellular carcinoma, which is the most common type of primary liver cancer. This graph here shows that at 3 years, patients with liver stiffnesses greater than 4.7 kilopascals have an HCC incidence rate of 42.7%, versus those with liver stiffnesses less than 3 kilopascals have an HCC incidence rate of 15.4%. And a very useful method for measuring this liver stiffness is through magnetic resonance elastography, or MRE. MRE is a non-invasive technique that can detect changes in mechanical properties in order to diagnose liver disease using shear modulus. So it is an extension of MRI, and looking at how MRI compares to MRE. If you have a healthy and a diseased liver, MRI is able to show water diffusivity as well as structure and tissue density of the liver. So while this can be valuable, MRE has an advantage in that it has the ability to superimpose the deformation of the material onto MRI images while vibrating the tissue. And this allows it for determination of how deformation changes with time. So you're able to get the actual stiffness values, as well as it can improve the sharper borders for regions of higher stiffness. However, there are some limitations in that MRI does not measure stiffness values directly, and it does require a high frequency of vibration for measurement. Additionally, more factors can impact liver tissue stiffness, such as frequency. So liver displays dispersive properties, meaning that it is dependent on frequency. Shown on the right, you can see 2 graphs from 2 different rat livers, one with stage 0 fibrosis and one with stage 3 fibrosis. So you can see that in addition to having very different storage moduli values, they also show different trends. And this could lead to issues in accuracy when using MRI. So for this reason, MRI requires calibration, which is done using something called an MRI phantom. So phantoms are materials that are used to calibrate a device. Traditional MRI phantoms, such as gels shown here, are pretty expensive and lack the complexity associated with human tissues. Additionally, it's important for a phantom to be approximately the size of the tissue you're looking to measure, which can provide a challenge given how large the liver is. So there is a need for a cheaper, easier to manufacture material to act as a phantom for liver tissue. So this summer, 2 materials that we were looking at were tofu and agar. So first, looking at tofu, tofu is a promising candidate for use as a phantom, as it has tissue mimicking qualities that are similar to that of some human tissues. It's also easily accessible, and it's far less expensive than some of the gels I showed previously. We also looked at agar. Agar is a common material for bacteria culture. It's very inexpensive, and it does have similarities to soft tissue. In addition to that, it is moldable, meaning that it can be poured into a container as a liquid and solidify into a solid, which can be great in the manufacturing process and is a large advantage. However, the mechanical properties of these materials are widely unknown, which is what we were looking at this summer. So to study the mechanical properties, we constructed a torsion pendulum shown here, which was made up of a rotational motor, 2 aluminum plates, as well as a lab jack. This summer, we added a ruler and magnets and weights. The ruler shown with the blue arrow, the dark blue arrow, and magnets and weights shown with the lighter blue arrow, in order to alter the frequency of the upper plates, as well as by changing the moment of inertia. So we were able to do a frequency sweep and change the frequencies without changing the impression of the sample. So here you can see an example of how we would collect this data. Shown sandwiched between the 2 plates is Trader Joe's super firm tofu, just from local Trader Joe's. And what's happening is I'm applying force to the ruler, and the tofu is oscillating underneath the plate. And we're getting data out of this that looks something like this. So what we're looking for out of this damped sine wave would be the oscillation angle to find the measured period and the amplitude. And we can actually use these values to find the storage modulus, which would only require the angular frequency, moment of inertia, form factor, and logarithmic decrement. So one thing that is very nice about the storage modulus calculation is that it does not need the magnitude of the amplitudes, meaning that various levels of force applied to the ruler or the upper plate during rotation do not change the values of storage modulus calculation. So what we would get out of this is we saw that super firm tofu does exhibit some frequency dependence. So you're seeing the storage modulus is increasing as the frequency increases. And this was over a range of about 0.5 hertz to a little under 1 hertz. And we were actually able to confirm this using a commercial rheometer. So the commercial rheometer is shown in blue and the torsion pendulum is shown in orange. And you can see they have very similar trends and are about in the same ballpark of values. These slight differences could be attributed to various factors, including just differences in compression level when performing the measurements. In addition to that, we were able to look at a larger span of frequencies using the commercial rheometer. So this is from 0 to 10 hertz, and you can see a large frequency dependence of the tofu over this range. So we also were able to look at agar, and you can see here that agar also has a large frequency dependence. This is over a range of about 0.6 hertz to a little over 3. However, when we did try to use a commercial radiometer on the same values, we did see a larger discrepancy. And it is kind of odd that you see around 0.5 hertz, the data seems similar, and then it seemed to diverge as frequency increases. This could be due to a variety of factors, including different samples, size and shape being different, or different compression levels as well. And it's something we'd like to look into more in the future. We're also able to compare these values to the rat liver rheology I showed previously. So on the left, you'll see that on the left, you'll see that agar actually mimics the rheology of fibrotic rat liver, very similar, like the trend very well. There are large differences in the G prime values, but it does have a strikingly similar trend. And then on the right, you'll see that tofu has a somewhat similar trend to those normal liver trends that were shown with the rat liver. So in conclusion, tofu and agar both exhibit frequency dependence similar to the dispersive properties of liver, which could suggest that they could be used as a tissue mimicking phantom to calibrate and validate MRE results in the future. Some of the stuff we'd be interested in looking at in the future is examining a wider range of frequencies using the torsion pendulum, as well as developing models to better understand the tofu and agar rheology. So in conclusion, I would like to acknowledge my mentors, Drs. Dawei Song and Dr. Daniel Iwamoto for their support, as well as Dr. Paul Janmey and the rest of the Janmey Lab for answering all of my questions, and as well as the CEMB, specifically Annie, for everything she's done for us, as well as Penn Engineering and the NSF for helping to fund this project. And with that, I'll take any questions.
Yeah, I have a question again. Yeah. This is Dennis. Yeah. You can hear me okay?
Yes.
Did you see that when you rotate your homemade rheometer that the tofu was strained at least at the surface that was rotating as expected? In other words, you had good coupling between the plate and the tofu. There wasn't slip.
Oh, yes. So to minimize slip, we did use sandpaper, and we did try and make sure there was full contact between the plate and the sample. There were some cases where at the lowest compression, or the highest frequencies, we did see some slip, and then we were able to redo those to make sure that we weren't seeing slipping. But yes, using sandpaper did minimize slipping as much as possible.
And so you somehow have a fiducial marker that really shows there's no slip. Yeah.
I'm not sure we have an exact marker, but that could be something we look at in the future to make sure there's no slipping. Yes, thank you.
Thanks a lot. Bye.
Could I ask a question as well? It's very, very interesting and obviously has a lot of outreach potential, but thinking of the work that you did you think about, or did you try putting any inclusions into the tofu or the agar and see what impact that had on your measurements?
We did not have a chance to do that this summer, but I think that would be a really interesting project to work on in the future to see how that would impact values.
Thanks.
Yes, thank you so much. Awesome. Are there any other questions?
All right, if not, thanks, Kat. Our next speaker is Zina Helal from the University of Texas at San Antonio, and she'll be talking about the work she did in the Wells Lab.
Okay, is my screen being shared okay?
Yes.
Okay, great. So good afternoon, everyone. Thank you so much for having me. My name is Zina, and today I'll be talking about varying the
mechanics and structure of polyacrylamide pillars as a model for the bile duct. And this summer, I've had the pleasure to work closely with my mentor, Dr. David Lee, in the lab of Dr. Rebecca Wells. Okay, there we go. So the bile ducts are tube-like structures that are part of a larger system called the biliary tree, which is a system of various types of ducts and organs, and it includes the liver, gallbladder, and pancreas. So the biliary tree primarily functions in production, storage, and transport of bile fluid. And bile ducts in particular transport bile from within the liver where it's produced to the small intestine, where bile has essential functions in digestion and absorption of fat-soluble vitamins. It aids in the removal and drainage of toxins through excretory pathways, and it also aids in metabolic regulation of hormones. So biliary atresia is, BA for short, is a rare but deadly disease that most commonly affects newborn babies, in which the bile ducts become backed with bile fluid and it's obstructed. And if left untreated, this condition can actually lead to liver fibrosis and liver failure very early in life. So there's a lot that's still unknown about the BA pathogenesis, but it's thought that as bile accumulates, ducts become mechanically stiffer. Additionally, associated obstruction causes drastic non-homogeneous bile duct curvature, and we can see this in this image here that looks at bile duct tubes that are of normal conditions compared to disease conditions, where inflammation and scar tissue has caused thickening and thinning of what normally is a smooth edge tubular structure. So both this increased stiffness of fibrotic ducts and various curvatures are thought to be key factors affecting possible cell behavior related to possible disease mechanisms and disease development. So with that in mind, the question of interest related to my summer project is, how does varying curvature and increased stiffness associated with development of fibrotic ducts influence cell behavior? And one way I was able to investigate this question in my lab was by building a physiologically relevant in vitro 3D model of the bile duct using polyacrylamide hydrogel molded into the shape of pillars. So polyacrylamide is a commonly used biocompatible hydrogel that is optically clear and has tunable mechanical and structural characteristics. So I used polyacrylamide to create micropillars with various stiffnesses and curvatures to model the tubular structure of the normal and diseased bile ducts. So the process of creating these micropillars involves 4 main steps, which I will briefly describe in the next coming slides, but it begins first with the fabrication of an SU8 silicon mold that we use to be able to generate and control the shape and curvature of the pillars, a modified polyacrylamide gel fabrication process, casting of the micropillars from the molds, and then the use of these micropillars in cell studies. So the fabrication process involves creating micropillars from a mold pattern transfer process, which first requires fabricating an SU8 photoresist mold that's embedded with our desired pillar geometry. And this is something that we're able to fabricate in the same nanotech center here at UPenn. So SU8 photoresist is a very viscous polymer solution that we layer over a treated silicon wafer. And we actually layer this SU8 at a thickness that's the maximum thickness that we're able to polymerize this type of polymer. So after that, we then expose the wafer to a UV light under a photomask, and the unpolymerized regions of the SU8 wash away during a development period, and that reveals a pattern of holes embedded into this polymerized SU8 layer. And here we can see that the holes generally have either a diameter of 75 micrometers, and we also generate wafers that have holes that are 300 micrometers in diameter, and they have a depth of about 500 micrometers. So this is an example of one of the wafers that we make, and you can see we have several mold patterns, all which are the same, but they have each about over 500 holes that we then use to generate the gels. So after we have our molds, we fabricate the polyacrylamide gel from ratios of acrylamide and its cross-linker bisacrylamide. And traditionally, these ingredients undergo cross-linking using initiators known as APS and TEMED. But for our purposes, we actually use a different cross-linking method using a photoinitiator. And so by varying the ratios of acrylamide and bisacrylamide, we prepare gels of stiffnesses ranging from 1.1 kilopascals to 14 kilopascals, which resembles soft, normal, and obstructed bile duct stiffnesses. In our gel, we also included red fluorescent microbeads that aid in later visualization of the gel using fluorescent microscopy. So finally, for the casting of the micropillars, a treated cover slip, as you can see here, is placed over a gel solution on top of one of the mold patterns, and it undergoes photo-initiated polymerization. And then it's removed, revealing an array of pillars. So this step of the fabrication process included optimizing some of the most challenging aspects of generating these micropillars. And some of these included first determining the best photo initiation exposure time, which we were able to optimize by systemic testing of polyacrylamide polymerization under UV. Another challenge was just the physics of getting a slightly viscous gel solution deep enough into such tiny holes to be able to later generate these tall pillars. And we were able to optimize this by combining dilution, vacuum, and sonication techniques. And then another big challenge was the removal of these cover slips from the mold without breakage of the polymerized pillars. And one way we were able to overcome this was by applying certain air and water incubation times after the gel has been polymerized. And that was something that helped keep the gel intact. So with overcoming these challenges, we were able to generate cover slips with an array of polyacrylamide pillars that had, again, diameters of 75 micrometers and 300 micrometers, and with stiffnesses ranging from 1.1 kilopascals to 14 kilopascals. And the way that we were able to verify the dimensions of our pillars was by taking confocal and epifluorescent microscopy images. And here, these are images from a confocal microscope, and we can see from the X, Y, Z orthogonal planes that we have a pillar. This is from the top view of the pillar where its diameter is 75 micrometers. And looking at these two images, we can measure the height to be about 400 micrometers, which is very thin and very tall. So now with these pillars serving as a relevant in vitro model of the bile duct, we can proceed with seeding the pillars with human gallbladder fibroblasts, which are HGBF for short. And these are cells that are commonly found in the extracellular matrix of the bile duct. So first we functionalize the pillars with collagen to allow for proper cell adherence. And we actually use a modified upside down cell seeding method. And this is a technique that we optimize so that cells are able to interact with the length of the pillar for a longer period of time, rather than cells sinking to the bottom of the gel over time, which would happen if we were to seed the cells upright. So cells essentially are suspended in media and allowed to interact for a period of 4 hours or so, and after which they're incubated overnight. And our hypothesis was that after letting cells attach overnight, cells will respond to stiffened pillars and pillars with higher degrees of curvature with changes in their phenotype and alignment in comparison to softer gels or larger gels. So with preliminary imaging, we're able to, and fluorescent microscopy and bright field imaging, we're able to see that we do have pillars that seed on the cells, and they do in fact respond, or we do have cells that are seated on the pillars and are sensing and responding them. So this video is a set of images that are taken moving from the bottom of the pillar to the top, and this red here indicates a presence of bead of the gels, and then the cells here are shown in gray. So moving from the bottom Z stack to the top, we see cells that are adhering alongside the cell, which is indicated by these grooves, but they're also seated on the bottom floor of the gel and as well on the top of the pillar. So this imaging was great positive data, preliminary data to suggest that our model can be used for continued cell studies and further characterization of cell response. So with subsequent confocal imaging of pillars, we were able to see the adherence of cells on the sides of the pillars, where here the gel is again shown by these red beads, and the cells are here stained in green. And so with this image, with this video, we can see a view going from the top down alongside of the pillar. And what's interesting to notice here is we can see the cells that are on the sides of the pillars, but we can also see these really extended protrusions of the cells, which is a sign of cell spreading in response to stiffened substrates. Another thing that we observed here was the alignment of cells along the pillars, which was something that we were interested to look into, but we can visualize better in these two bottom images that show just the distribution of the cells without the noise from the red beads, that the cells are almost aligned in a way that is up and down alongside the pillar. And we can really see this one cell that's just extending all the way, almost the entire length of the pillar. And we see that trend across this specific pillar. In the future, we will continue to compare cell alignment of thinner pillars and pillars of different stiffnesses to further characterize the molecular mechanisms underlying cell response and possible modeled fibrotic tissue conditions. Other things that we can also continue to explore in addition to further quantification of alignment and phenotype, and by phenotype that can include looking at quantifying exactly how spread out the cell is by looking at its surface area. But additional things is we can look for signs of cell activation or differentiation into from fibroblasts into myofibroblasts. Another thing that we can include in our model is the inclusion of TGF-beta, which is an inflammatory cytokine. And by including this, we can investigate cell response in normal conditions and then inflammatory conditions. And then we can compare the two and see how that may be related to possible disease mechanisms as well. We can also investigate we can include additional cell types in our model, such as other extracellular matrix proteins that are known to be surrounded in around the bile duct, and also collagen and hyaluronic acid. And as of right now, we are modeling some of the smallest structures of the bile duct, but with our model, we can also expand it to larger structures of the bile duct as well. Overall, being able to optimize these polyacrylamide pillars allows for physiologically relevant modeling of the bile duct, which is an avenue to further investigate and understand molecular mechanisms that may be underlying disease and conditions that we're still trying to understand, such as biliary atresia. So with that, I'd like to acknowledge and thank my mentor, Dr. David Lee, and the lab of Dr. Rebecca Wells for helping me throughout my research project, and obviously this research would not have been possible without them. I'd also like to thank students of the Wells Lab and Annie for helping put together this amazing program in the CEMB community. And with that, that concludes my presentation, and I'm happy to take any questions. Thank you.
I think we'd have time for one question if there are any. All right, if not, thank you, Zina. It was great work.
Thank you.
Our next speaker will be Emily Rudolph from Widener University, talking about her work in the Janmey lab. And we can see your slides, but we can't hear you.
Can you guys hear me?
Yes.
Okay. Sorry about that. Okay, I'm just gonna start over. So again, hi, my name is Emily Rudolph. I am a part of the Janmey lab this summer, working with Banaz under creating an electrically conductive scaffold used for mechanobiology, or more specifically, tissue engineering. So the kind of background for this project is there are not a lot of electrically conductive scaffolds out there. Chitosan is an example of a not electrically conductive scaffold that we use in this experiment. We decided to use chitosan because it is a promising biomaterial in tissue engineering. And so we decided we want to use it. The reason we wanted to create an electrically conductive scaffold is because stem cell behaviors change because of an electric stimulation. Electric stimulation causes stem cells to change their proliferation, differentiation, as well as their morphology. So creating an electrically conductive scaffold would help facilitate the electrical stimulation to the stem cells. So we had two main ideas in this study. The first one is to create an electrically conductive scaffold. Like I said, there's not a lot of electrically conductive scaffolds out there. So we decided to combine chitosan with polyaniline. Chitosan is not electrically conductive, but it is a promising biomaterial, like I said, but polyaniline is electrically conductive. So we combined the two of those to create an electrically conductive scaffold. Our next part of the experiment was to culture cells on this electrically conductive scaffold. We decided to use human mesenchymal stem cells and evaluate their behavior. The behaviors we focused on were morphology and viability of these human mesenchymal stem cells. So again, just walking you through the process, we created a conductive substrate. We combined non-conductive chitosan with conductive polyaniline and created a conductive scaffold. We also just made a chitosan scaffold to compare it to. Once we create the scaffolds, we characterize them. We use the FTIR machine to look at the chemical bonds in the different scaffolds, which I will get into on the next slide. We also measured their electrical conductivity because we knew chitosan and polyaniline scaffold was electrically conductive, but we didn't know how electrically conductive. So we wanted to measure how much electrical conductivity this specific scaffold gave out. Once we characterized the scaffold, we cultured the cells and applied electrical stimulation to the cells. And we did a viability assay as well as took images of the cell and analyzed them, which we will get into on further slides. So this is just our scaffolds characterization. We did an FTIR measurement. So the top green line is the chitosan and polyaniline scaffold. So as you can tell, the green line has some similarities to the blue line, but it also has some differences. The differences is the polyaniline shining through, and I put red arrows to highlight the different points at which the peaks differ. And the red arrows correlate to the specific wave number, which in return correlates to the specific chemical bond I have listed in the table. We also did an electrical conductivity measurement, which again, chitosan is not electrically conductive. However, combining chitosan and polyaniline together creates this electrical conductive scaffold. And it's actually really interesting because this amount of conductivity approaches the level of conductivity a tissue needs to where a stem cell would differentiate. So this could possibly give leeway to new experiment and project ideas. So again, once we created the scaffolds, we cultured cells in them and applied electrical stimulation to them for 20 minutes, one time a day. And the cartoon I just have showed is just the electrical stimulation being applied again for 20 minutes. And so here are the results we got after the electrical stimulation. We did a viability assay, which we did after 24 hours, 3 days, and about a week. And so over time, you can tell that we did the control, which the control was just no scaffold, just the cells growing on a plate without a scaffold. And then we have the non-conductive scaffold, with the conductive scaffold. And you can tell over the week that there's no major difference in the viability of the cells from the electrically conductive scaffold in comparison to the non-conductive scaffold. There's no major difference. This shows that the chitosan and polyaniline scaffold has good biocompatibility. Here are the images we took of the cells after we stained them. We stained them about 3 days after, and again, we applied electrical stimulation to them once a day for 20 minutes. And then on the third day, we stained them and took images of them under a microscope. We stained the cytoskeleton, which is the main, where we gathered the main data from. We took the aspect ratio, which is just the longest side of the cell over the shorter side of the cell. And what you can tell from that data is just the size of the cell. So if you're looking at the pictures, the control is they're round cells. In addition, the chitosan cells also very round, but the chitosan and pani are very different. The electrically conductive scaffold did initiate some kind of change of shape. As you can tell, the cells are very elongated. And then we have data to support what we're seeing here. The aspect ratio was recorded. And as you can tell from the data, the control and the chitosan are have very similar aspect ratios, showing that they are round. However, the chitosan and polyaniline cells have higher aspect ratios, showing that elongation we are seeing in the F-actin stain picture. We also recorded the surface area of the cells. As you can tell from the graph, the control and the chitosan samples are very similar. However, the chitosan and polyaniline cells have an increase in surface area, and that's just showing you what this electrically conductive scaffold with the electrical stimulation increases the surface area as well as the elongation of the human mesenchymal stem cells. So just to wrap everything up, what we found in this study is that the chitosan and polyaniline scaffold is significantly more conductive than just the chitosan scaffold. The chitosan scaffold is not conductive. However, adding polyaniline to the scaffold makes it very conductive, and the level of conductivity approaches tissues such as brain tissue and cardiac tissue, which is really interesting and what can be done on further studies. We also found that the combination of electrical stimulation and this electrically conductive scaffold shows good biocompatibility, as you saw with the viability assay. And the human mesenchymal cells also became more elongated and increased surface area due to the electrically conductive scaffold being initiated with electrical stimulation. So I just want to thank everybody in the Janmey lab, as well as the CEMB program and Annie for giving me this opportunity and this great summer of doing research. If anybody has any questions, just let me know. Thank you for your time.
Great, thanks, Emily. Are there any questions for Emily? I have a quick question. You probably covered this, but does the addition of the chitosan, I guess, change any other properties of the scaffold or anything?
So what we could tell in the experiments we did, the chitosan didn't really change anything because we did just the chitosan scaffold. And that was pretty similar to the control, which was no scaffold, just the cells growing on the plate. So the chitosan really didn't impact anything in what we measured. However, maybe in another aspect that we didn't measure.
Sorry, what about the polyaniline?
So the polyaniline made the cells more elongated. That made it, well, the polyaniline made the scaffold electrically conductive, which facilitated the electrical stimulation, which made the cells elongated and the cells increase in surface area.
Okay, great. Thank you.
Thank you.
All right, so our next speaker will be Elaine Nagahara from Johns Hopkins University. She'll be talking about the work she did with the Wells and Stebe labs.
Are you able to see my screen?
Yes.
Okay, that's good. Thank you for the introduction. So yes, today I'll be talking about the work that I did this summer in the Wells and Stebe lab on understanding the role of microenvironmental curvature on cholangiocyte behavior. So I just want to include here a brief overview of what I'll be covering today. And I first want to start off by setting the story a bit with why I studied what I did and the basic principles behind the system I developed. So conventionally, cell studies are done on planar surfaces, but the cellular environment is not just a vast expanse of planar geometries. In fact, it's quite geometrically complex with a variety of curvature. And in the body, we have different organs that have distinct curvatures, such as sweat glands or sebaceous glands that have this more bulbous spherical-like curvature shape, as well as blood vessels or bile ducts that, when branched, you can see that they have this unique saddle-like region here that generate distinct curvature. And because of the lack of many non-planar cell studies, we really don't know too much about how cells respond to curvature. So the goal for this summer was to study how curvature affects cell behavior, specifically focused on cell migration and morphology. And here we use cholangiocytes as the cells of interest. Cholangiocytes are epithelial cells of the bile duct, and because they naturally form curved surfaces in the body, they served as a physiologically applicable candidate for this study. Now, in our models, we wanted to capture something called Gaussian curvature. And in differential geometry, this can be defined by an equation you see here on the left. So it's the product of the principal curvatures at a specific point. The principal curvatures are the maximum and minimum of the normal curvatures. And here they're denoted by kappa 1 and kappa 2. And you can see here in a couple example surfaces. And different from curvature that you may typically think of in your everyday life. For example, you may think of solids like cones or cylinders as being curved and having curvature, which they do. But in terms of Gaussian curvature, they actually have 0. And that's because you can see here in the example on the right, you can see one of the principal curvatures has a value of 0. And because it's a product value, if one of them is 0, the whole thing goes to 0. Now, one important thing to consider when you're talking about Gaussian curvature is the sign. And because it's a product value, you could probably guess that the sign depends on whether the signs of the principal curvatures are the same or whether they're different. So for positive curvature, if the two principal curvatures have the same sign, then you achieve this. And if for negative curvature, the signs have to be opposite. And this results in distinct shapes for the respective types. Now, this is an image of that shows how we incorporated this Gaussian curvature, both the positive and the negative, into our models. And while some past studies have looked at the effects of curvature on cells with stiff substrates, this is really the first time we're looking at the effects of curvature with softer substrates. And this is an important step forward because we know that stiffness can have a great effect on how cells behave. And here we use collagen, a biologically relevant materials, to create a soft environment that cholangiocytes or cells typically experience in vivo to achieve these controlled model microenvironments to culture our cells on. And before moving forward, I just want to emphasize, I think the easiest way to distinguish between, for now, the difference between positive and negative Gaussian curvature is to think about the shapes that they're represented by. So for positive, you can see here where I've outlined at the top, where it says positive cap, they tend to be represented by more dome-like spherical shapes, whereas negative Gaussian curvature, you'll typically see a more saddle-like shape here. Here I've labeled at the bottom. I just have a couple schematics fitted here to hopefully give a better idea of what this would look like in 3D, because this is more of a cross-sectional view of the image. So moving on to how I approached this project. So there were two types of models that I created with two different sizes. So one were made with glass particles that were about 2 millimeters in size. And then the other one were smaller. They were polyethylene particles that were about 150 to 180 microns in size. And these are both spherical structures. And here's a brief overview of the fabrication procedure for both of these. They differed a bit depending whether they were the larger particles or the smaller ones, but they had the same basic principle behind how they were created. And so how that was done is I placed the particle in the center of a petri dish, and then essentially I would put collagen on top of the bead. And as the gel flowed down the sides of the bead, it formed this nice skirt. And that's what I was referring to earlier, is with the negative curvature there and the positive curvature at the top. And this curvature was maintained also by capillary action, which acts in opposition to gravity to allow the gel to move up the sides of the particle due to surface tension forces. And finally, we then seeded cells onto these models and cultured them for about a week before imaging. Now moving on to the results, I broke this down into two sections. So the first part, I want to talk about the characterization of the model itself before I go into talking about what we saw with the cells. So for these systems, we were able to gather measurements on some of the angles here. And these contact angles can give us an idea of how strong the curvature is. And this will also then allow us to maybe later in the future adjust the curvatures, how strong they are with things like substrate treatment to vary the wettability of the collagen gel or the collagen volume, etc. And here we measured the particle collagen contact angle, which essentially describes how much the collagen likes this particle, how closely it can hug to the sides of it. And the collagen substrate angle describes how much the collagen likes the substrate and how much the collagen can spread on this surface. And in general, a smaller angle generally can suggest that the strength of the curvature is larger. And here I just want to quickly include a graph of the curvature of the contour from this goniometer image that you kind of saw a similar one earlier. And the main takeaway here is that you can see that there's both a positive curvature region and then a negative curvature region here with an inflection point where you see the transition at 0. And while we technically need more information to calculate the exact value of the Gaussian curvature, since this is more like a two-dimensional cross-sectional image, we know that this model is roughly axisymmetric because of the spherical nature of the particles, which serves as the foundation of the system. So we can assume that the direction of the other principal curvature, and we can see that we have not only positive curvature, but also negative. So now moving on to what we saw with the cells. So this is with the larger model here. Are you seeing, sorry, my slides might have switched a bit. Okay, sorry, the larger model here. So after seeding the system with cells, we saw protrusions growing in the direction of the spherical cap towards the top where the positive curvature is, migrating at least 250 microns towards it, assuming that the cells were originally at the bottom due to their tendency to settle there. And the cells towards the top cap had an aspect ratio of 32% higher than the cells towards the bottom and on a flat surface without the curvature, implying that the cells towards the top experienced a greater degree of deformation. And we also saw a similar trend with the cholangiocytes on the smaller models. They also migrated a total distance of about 130 microns towards the top of the model where the spherical cap is, and they had a 38% higher aspect ratio than the cells towards the bottom. And cholangiocytes are, again, they're epithelial cells, the bile ducts, and as epithelial cells attach and they move, they tend to become less circular and more morphologically fibroblastic. So the increase in aspect ratio for the cells here towards the top supplements the idea that the cells may have needed to form more adhesions to maintain their position on like that slope, as well as to form protrusions to migrate. So in conclusion, we were able to create models that studied how Gaussian curvature affects the behavior of cells And here it was revealed that cells seem to migrate towards the top of the cap where the positive curvature was, and that the morphology differed depending on the location. And we can use this information to control organization, cell organization models in the future, even for perhaps like drug testing models that can be made gland specific by tuning the magnitude of the curvatures or the localizations of them to emulate the curvature that you would naturally see in the body. And one limitation that I would love to explore further in these studies is that we don't know whether stiffness played a role really in influencing the cell behavior, because towards the top, as you go towards the top of the model, the layer of collagen coating the particle does get thinner. So that is one other factor that we have to explore further. And one way we could do that is by creating models, creating these molds to directly cast the collagen in instead of having like the particle then the collagen. That way, instead of having a gradient of varying stiffness, you have uniform stiffness throughout the whole system. And finally, I think exploring how other cells respond to this in the future would also be interesting to see if there's similarities or differences in their behavior. For example, see even how curvature could induce branching, again, in cholangiocytes or even blood vessels in angiogenesis. And I just want to conclude by thanking Dr. Wells and Dr. Stebe for giving me the opportunity to work on this really exciting project. My amazing mentors, Dr. Gupta, Mrs. Zaya, Ms. Dang, for teaching me so much throughout this whole summer. And finally, none of this would have been possible without the funding from NSF and the CEMB. And thank you, Dr. Jeong, for coordinating this summer. And I'd be happy to take any questions.
All right, well, if there aren't any questions, thank you, Elaine. That was very well done. I think I finally understand Gaussian curvature from your presentation. And so for our final presentation today, we'll be hearing about the work done in the Hughes Lab by Marysol Chucardi from the City College of New York and Michael Foster from the Community College of Philadelphia, soon to be Temple University.
Thank you, Annie. Hold on a second. I'll share my screen. Can we see it okay?
Yep.
Okay.
Alrighty, hi everybody. So this summer, Michael and I got to work in the Hughes Lab and we investigated the mechanical basis of kidney branching morphogenesis. And kidney branching morphogenesis leads to the formation of the urinary collecting system. And it begins when the Wolffian duct invades the metanephric mesenchyme. And this invasion into the surrounding mesenchymal tissue will lead to the development of a tree-like structure. And as this process is going on, the cat mesenchyme cells will epithelialize and mature into nephrons, which are located in the armpit region of the branching tips. So for this branching process, there's also two important signaling molecules. The first one is glial cell-derived neurotrophic factor, or GDNF. And this is present in the cat mesenchyme, shown here in the red. And this binds and activates RET, which is located in the ureteric bud, shown here in the blue. And as these RET-activated tips branch out, they are also more motile and responsible for the branching, whereas the trunk cells are less motile and responsible for structural support of this ureteric tree. So an abnormality in the developmental process can cause many issues in the filtration system. One of them is ureteropelvic junction obstruction, which blocks the flow of urine into the bladder. Another example is renal agenesis, which is a lack of one or both kidneys. And finally, horseshoe kidney, which occurs during fetal development, and it's when the base of the kidneys fuse together. So we know that cell behavior is not only affected by chemical signaling, but mechanical properties can also affect how the cells behave. And so because of this RET activation increasing cell motility, we wanted to investigate if RET activation would also affect the forces exerted by these ureteric bud-like cells. and see how branching morphogenesis of the ureteric bud would change the mechanical environment during kidney development. And we did this using traction force microscopy.
Now, cellular traction forces are generated by adhered cells onto either substrate or onto extracellular matrix. These traction forces play a pivotal role in the motility and the migration of cells during important stages like development. Cells are able to adjust proteins such as actin filaments for structural stability and generate proteins called focal adhesions to allow themselves to be adhered to the extracellular matrix of substrates. Now, traction force microscopy helps us study these traction forces that are generated onto substrates. We use things like hydrogels that have fluorescent microbeads inside to act as markers so we can track the forces generated by these cells. Okay. There we go. Now, we use Madden-Darby canine kidney cells, which are a renal cell line that when transfected with RET, it makes them a good model to study RET activation. We transfect these cells with RET receptor to understand how the ureteric bud cells will act during process of branching morphogenesis. Excuse me. RET transfected MDCK cells have been studied and observed to have increased motility and migration in the presence towards sources of GDNF, which is similar to the ureteric bud cells during the process of branching morphogenesis. Now, the reason why we use polyacrylamide gels, play my video, is because they have two very important properties. The viscoelastic property of polyacrylamide gel, allows for the forces from the cell to cause deformations to the gel, so that with the assistance of these fluorescent microbeads that we put inside of our gel, we can observe these traction forces accurately. Now, this accompanied by the linear elastic property of a polyacrylamide gel, which is linear in proportion to the stress and strain that the cell generates onto it. We can measure these forces accurately given the linear elastic property with the fluorescent microbeads when they're pulled by the cell and when the cell is released from the gel. These beads move back to the initial position upon detachment, which allows for analysis to be precise.
So our project could be broken down into 4 main steps. The first one, we fabricated a polyacrylamide gel, and we tested different heights of the gel to take into account the working distance of the microscope, as well as different densities of the fluorescent beads so that we could visualize individual beads under each cell that was attached. Next, we promoted cell adhesion by adding fibronectin to our gel, and we did this using sulfosampa as a protein crosslinker so that it would indirectly bind the fibronectin to the polyacrylamide gel. And we plated 2 different groups of cells, one in basal media, which was our control, and the next one with GDNF GFR alpha 1, which activates RET. And finally, we used a confocal microscope to image the gel before and after cell detachment. So we had an image of a stress gel as well as a relaxed gel.
Now, when analyzing these 2 images, when analyzing our images, we've been using ImageJ. 2 important plugins that we have been using is to track the cellular traction forces are PIV and FTTC. PIV is used to determine the displacement of these beads, which appear as a vector map shown right here. And this image is then used to generate a PIV displacement map, which is correlated to the amount of beads used and the quality of bead tracking, which is right here. Now, the FTTC plugin is used to reconstruct the cellular traction force pattern based on the algorithm of the program, where the measurement of the traction force is in Pascals, which are then converted to Newtons, which we do. In order for us to successfully run these programs, we have to align our images with the stress and the relaxed gels. And following alignment, we run the image through the PIV first, which generates a displacement map, as you can see. And then it also generates a code that we can put through the FTTC plugin, which generates our force map and gives us our calculation of the traction forces that our cells have generated onto our gel.
So before we talk about what exactly we found, I wanted to show you guys what our cells looked like under the confocal microscope during detachment. And as you can see, it may not actually be so noticeable, but as a cell detaches, the beads are actually moving into their original state. And so we took the first time frame of this video as well as the last time frame, and we then paired the 2 images together and ran them through this published software. And so as Michael mentioned, the PIV plugin gave us this displacement map and text file. And then we used that information as well as the Young's modulus of our polyacrylamide gel and the area of our window to determine a traction force value. And an important thing to mention is that the Young modulus of our gel was actually 850 to 900 pascals, which is very similar to that of soft tissue, which is about 1000 pascals. So as we can see here, the GDNF group had higher traction force in the leading and trailing edges of our of the cell of about 1.28 times 10 to the negative 17 newtons. And so we actually averaged both our groups together to see if there was a difference between the forces that these cells exerted. And after doing a t-test, we found that our preliminary data did show that there is a significant difference between cells that were reactivated versus non-reactivated. So just to give a brief summary of our whole project. So we were able to investigate the mechanical difference between MDCK cells that were reactivated versus non-reactivated using traction force microscopy, and specifically visualizing the bead displacement. And we found that GDNF cells, so reactivated, actually did exert a greater force on this polyacrylamide substrate. So our data may contribute new insights into the mechanical effects of GDNF signaling in the developing kidney. So in addition to knowing that GDNF can increase cell motility, it could also be mediating intracellular forces as well as remodeling the extracellular matrix as seen by this increased traction force. So a next step would be to determine the traction force in primary ureteric blood cells, because in the future, by fully defining the changes in the microenvironment of the developing kidney due to this branching morphogenesis, we may have some more information as to how the kidney cells differentiate. And so we just wanted to thank Dr. Hughes for welcoming us into his lab, as well as John, Aria, and Nate for helping us throughout the summer with this project. And of course, Annie, because this program wouldn't be possible with everything that you've done for us. Thank you. And we're happy to take any questions.
Yep.
Great. If there are any questions, please unmute yourself or put it in the chat. All right. Well, if not, thanks, Marysol and Michael. And thanks to all of our speakers today. And congratulations, you guys all did a really, really outstanding job. And I think I'm always amazed, but with you guys especially, at just all the progress that you made, and not just in the projects that you presented, which were fantastic, but also just in your skills and your confidence and your independence that you gained as a researcher. So you guys should be really proud. I also just want to, before we wrap up, thank the mentors, the faculty, and the trainee mentors that worked with you all. I think this program really wouldn't be possible without them, to take you guys under their wing. And so they should also be very proud and celebrate this accomplishment too. And as a thank you, I do have t-shirts for the PIs and the mentors. So you'll hear more about that soon. I also just think that I can speak for the CEMB leadership in saying that, we're really proud that you guys are all affiliated with the CEMB now. And we hope that you'll stay with us, moving forward. We're really excited to see where your careers go and what other accomplishments you guys will have from now on. And so, again, congratulations to everybody. And I hope you guys had a great summer.